हेलो हेलो हेलो। हेलो हेलो उमन नहीं आवा था। हेलो आवा था। हेलो 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 हेलो। ये वो स्पीकर ठीक है यार, वहीं लगे बैठा रहते हैं, क्लियर सुना था। हेलो ये देखा यार, ज्यादा घी घाट वाले लगा थे यार, नहीं कर रहा थोड़ा ही समझ यही नाटक कर रहे थे यार, वाला आवाज खींच ली थी, सुना बतावा थे। ओके बंद कर। हेलो हेलो ओकर वजह तेज कह देबे। दरअसल का है कि नहीं, एकर कैपेसिटी है पॉलीकम वाला। हेलो हेलो
टाइमिंग के लिए जल्दी कर दिए।
मेल 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 वो ऑनलाइन वर्कशॉप से ही किया गया है।
हाँ
उनको बोल दो एक बार।
सर मेल में भी हाँ हाँ
हाँ टूट गया ये, टूट गया।
अभी 10 बजे से करना है।
नहीं कैसे हैं शशि जी?
रात
तो अभी अनम्यूट है।
पहुंची सर
अभी
बातचीत करें।
अभी अनम्यूट है कि नहीं है?
वो एक तो मेल कोई देखता नहीं है, इतना बार मेल कर रहे हैं वो देखे नहीं है। तो मुझे वो भाई 15-20 मिनट लगेगा क्या करें।
तो आप उनको सुबह रिमाइंड नहीं किए थे एक बार?
माइंड कैसा, मैं उनको भेजता हूँ कई बार।
हैं? ये बाकी लोग के इसमें कैसे आएगा कि भाई यानी मैं भी दिख रहा हूँ बाकी लोग भी दिख रहे हैं, कैसे हो पाएगा?
Hello.
Good morning, everyone. Mic should be on. So, all the participants again on the second day of this workshop. I hope that you had a good interaction on the first day. Definitely, you'll have a better interaction as we will interact more and more. And I hope that most of your queries were solved. And today we'll be having two important lectures. One is from Dr. Manas Kumar Naik. He is a scientist in the University of Iowa, and he'll be joining us soon. After that, Professor Kemparaju will join us from the University of Mysore. Both are having a very good research career, and Dr. Manas, fortunately, is product of our department only. He has after that he has done his post graduation and postdoc and other things in US only, and now he is settled there. And he is now a faculty, a scientific faculty in the University of Iowa. So he'll be interacting with us soon. And after that, Professor Kemparaju will interact with us. He has a very good hold on netosis and other techniques. And definitely, he is a very good researcher in venomous use of in use of venomous substances, and definitely we'll interact with him in the later session. After that, we'll have a session with Dr. Josna Kailashia, who is a faculty in our department, and she will guide us through the very important technique of flow cytometry. Also, it will be followed by a video for the same technique. So it is a virtual lab experience for all of you for flow cytometry. As flow cytometry is a very important and diverse technique, and also it has a very wide implication in different fields of science. So I hope that all of you will get benefited with that. And if you have any doubt from the previous session of yesterday, please feel free to ask. Today we'll be mailing all of you the YouTube link of this particular program, so that this will be a YouTube live, and all this session, all the presentation will also be there in the YouTube server, so that if you have any glitches in the reception or anything like that, or if you have missed any small session, or there was some timeout, definitely you can also join and again revisit this particular session through YouTube. So I guess that this will fulfill the demand for videos that few of the participants have raised yesterday. So I'll hand over to Das sir for his introduction.
Good morning, everybody. Now I'm seeing that there are 50 participants, and now it will become 51. And we expect more. But what happened that Manas, who is in USA, so he is reaching his lab. It is now 12 o'clock midnight. So he's reaching his lab to make the their communication facility on. So I think there's a little bit time gap. So I can address. I was listening to some of the queries and your concerns yesterday. So I think I can utilize this time to address, try to address at least some of them. Now I was very much impressed with Dr. Swatanka Prasad Madam, that from Jamshedpur. She is very much alive on the now at present also. And Madam was very concerned that we don't have an aggregometer, and then some these equipments. No, Madam, that you see that the whatever you are seeing in our laboratory, that has not been built in one day. We have started from the scratch, from just nil, nothing, with a simple centrifuge machine. So it doesn't require really to acquire an aggregometer to kickstart your research on platelets. Our main goal to motivate all of you to engage a part of your research, whatever area you are working can work, but part of your research in platelets and thrombosis. And we are very much well behind you in this effort. Now, regarding the lack of aggregometry, just whatever equipments you are planning to use or you will be using or listening, just understand the principle of this. Aggregometer means platelets in before activation, they are scattered in the suspension, light cannot pass it, transmitted through this. Once you aggregate, platelets get aggregated, they cluster together, so there is enough space for the light to pass through this. This is a simply transmittance or what is it called, turbidimetry type of thing, which is a very primitive traditional method in any laboratory, which has been digitalized and modernized and commercialized now. Equipment is around, I think, 10 to 15 lakhs, something, and we have purchased, we have 4 or 5 setups now. Okay, so now for that, madam, you can try a simple thing. Try to prepare a very good, that is a really challenging thing, to prepare a very good sample of platelets. Because yesterday, Sushil has told, Josna and other people have told to you how to prepare a very unstimulated sample of platelets. That is really challenging. And everybody has their own customization, every laboratory. You see different laboratories, you see the, read the literature in the paper on the platelets and see the method section, platelet preparation. And different laboratory has little bit this side, that side, they are tweaked the methods so that they get very much customized to their own lab, to which they are very much satisfied. Earlier, we are using aspirin. Somebody asked, yes, we are using aspirin because I started this research long back in 90s when I went to Germany for 2 years in a lab, which is very much specialized for platelet research. So whatever I acquired from the lab, I just translated into my own lab back here. So they are using aspirin. I don't know now they are using it or not, but they are using. And you see our old papers before 2020, all the methods we are using aspirin. Now, we have some difficulties, some good things, some at least we have published successfully. But then we did something experiment, there's some problem, then somebody told that, okay, you use some prostacyclines or PGI2 or PG1, something, and don't use it. So it is a hit and trial method which will best suit to your own laboratory and your centrifugation machine, your temperature, the ability, capability of your workers, so that you can have a very good preparation. Some people use albumin, because you can have literature too, which note qualities or positive, negative attributes of these different reagents. Albumin will make the platelets make the platelet less sticky. Okay, so they use
Yes,
albumin.
sir.
We don't use albumin. Now, once you prepare a very good preparation, you don't have an aggregometer. There are so many ways. Simple, madam, can I request you to try that? Try to stimulate the platelets. You have a microplate reader or colorimeter, just simple transmittance, optical transmittance. So 600 nanometer is a visible range. And in the 600 nanometer, you see that before just platelet suspension. It should be very opaque, buffer, 100%.
Yes, sir.
And then you aggregate, say over 2 minutes, 5 minutes, and while aggregating, you simulate a aggregometer in 37-degree, put the tube, and with the hand you can shake it like this. In the 37-degree, go on shaking. So if you have to do 5 minutes, do it for 5 minutes. Then immediately put it, and you see that how much is the light transmittance. Just it is a home exercise, and you can find, perhaps you can write a good paper, good literature for the laboratories who doesn't possess aggregometry. So then you can use your own reagents, some Ayurvedic preparation, which you expect to be antiplatelet, some this thing, that thing, some drug. And you can see that by this method, using very much manual method, but scientific method, I don't have aggregometer. And then
Yes, sir.
you can write a proposal and get a good machine. Yes, madam, you want to say something?
No, sir. Please continue, sir. I just
I am very much afraid yesterday that we should not scare the participants away, that they should think that, okay, we need this machine, then we do experiment. No, madam, it is the other way out. You should have very good idea, then you stretch your idea. Now, for example, I can suggest you another thing for all the participants I am saying. You can have a cover slip. Put get some fibrinogen, which is very inexpensive, or collagen, inexpensive. Just put a drop on the cover slip, let it dry, and add platelets on this. It won't cost you much, and platelets will start spreading. You can check it with any ordinary microscope, phase contrast microscope as well, and you can check your reagents. Okay, madam, now Manas has arrived at the other end, other part of the globe, and he is very much there, alive on the scene. So I request Kamalesh to introduce Manas.
thank you, sir.
Thank you. Thank you, Manas sir. It's great to see you again. I think I met Manas sir first in 2010 when I came for a workshop in B.H.U as a PG student, and then Manas sir was was a research scholar here. But again, years passed very quickly, and I see Manas sir here now. Okay, so Dr. Manas Kumar Nayak is a research assistant professor in Institute of Iowa. He has done PhD in biotechnology from B.H.U. And again, he has acquired research position in University of Texas, University of Iowa, and now he is continuing University of Iowa. He is different patents. I think 5 or 6 US patents. He has got many awards, and I'm having a very long list, so it will be difficult to read all of them. But and he has a very good index paper. Total citation is 1160, having H index of 19 and I10 index of 26. And he has a long list of publication, almost 32 out of them. And he is also reviewer in many of the very important peer-reviewed journals, including Blood, Scientific Reports, International Journal of Molecular Sciences, ACTA, Biomaterialica, and many others. And he is a editorial member editorial board member in International Journal of Molecular Sciences, current pharmaceutical design, Austin Internal Medicine, and Austin Journal of Nanomedicine and Nanotechnology. So I again welcome Manas sir for his talk. I know that it is a very odd time for him to have a, because of different timelines. I'm very happy to see him here again. Thank you. Welcome, sir.
Screenshot. Thank you so much. Am I audible?
Yes, sir. Please share your screen, sir.
Yes, sure. So how to, okay.
Yeah.
So you can see my slide?
Yes, sir. Yes, we are able to see you, sir.
So yeah, good morning to all. I'm working as a research assistant professor in UIOA, as Kamlesh just introduced me. And
Please maximize your presentation, sir.
sorry?
Please maximize your presentation. Please make it on a slideshow mode. Yeah. Yeah.
Is it okay now?
Yes, sir.
Okay. And I'm also an alumni of Professor D. Das laboratory in Sierra. So you can see my slide, right?
Yeah. We are able to see your slides, sir.
Okay, so I'm also alumni of Professor D. Das laboratory in B.H.U. I'm really thankful to Das sir and the event organizers Nitesh and Kamlesh for having me in this workshop. I'm really happy. I would like to tell you all the attendees and students that Professor D. Das laboratory is the most advanced thrombosis research lab in the country, equipped with all the state-of-art technologies under the one roof. You name the instrument and sir has all that. He is an eminent scientist who is always available for scientific discussions and also supports all his students even after leaving the lab. So I am really a very proud product like from the DDAS laboratory. So yeah, I'll start my presentation now. Is it going forward? Yeah. So I want to tell you really very quick a little bit about the importance about hemostasis before going to my data. So, which is the main focus of the thrombosis research nowadays. When there is a clot in the body, it really needs to be stopped very quickly, unless the consequences will be more detrimental. So the cause behind this is the blood system is a very pressurized system. If it doesn't stop bleeding in time, so there will be a very rapid blood loss, and it may end up with severe consequences. So we have a very complex system that is called hemostasis, works here in combination of the activated platelets and all other blood cells, and also immobilized fibrin strands. So it makes a plug there and seals that clot and heals that wound, and this is how it should happen. This is the normal hemostasis. But when if it happens outside the body, then it's good. If the same thing happens inside a blood vessel inside our body, which is bad, and you are in danger. So this is a normal artery. The normal artery should seen like this, but in a diseased artery, in atherosclerosis, the plaque is there, and when the plaque ruptures, gradually the lumen decreases, and when the plaque ruptures, there is a clot formation happens inside the blood vessels, and sometimes this blood vessel always occlude, and the blood flow beyond this point blocked. And so due to that, here you can see if it happens in a coronary artery in the heart, so it stops blood flow beyond that point to the myocardium. So it may happen like to anyone, even in the younger age to older age, and it causes the myocardial infarction or heart attack. And if that clot just cleaves away and loosen up and travels to the circulatory system, that's and reach to the brain and like very tiny arteries of the brain, then it stops flowing blood beyond that point and it causes ischemic stroke. So there are two phases of that hemostasis. One is platelet and other is the coagulation system. So I'm going to talk about the platelets today. So these are very small enucleated cells in the circulation and present in very high quantities, and they are very sensitive and really very responsive to the injuries. And for that, they transform themselves from the quiescent state to large aggregates. And activation of the platelets in like in response to different stimuli like collagen in the subendothelium happens, and this is how the signaling inside a blood platelet happens. So like this is a very complex signaling pathway. I will not confuse you all, so I'll just go a little bit slowly. There are different receptors present on the platelets, and to different stimuli like collagen and ADP, all other thromboxane, all the molecules, their respective receptors, and when they come in contact with their stimuli, then there is a signaling pathway starts, and that is called inside out signaling. And ultimately this alpha 2 beta 3 integrin that opens up and ready to bind with a fibrinogen, and after that also outside in signaling starts. So without confusing with this complex signaling pathway, I want to just go very with a simple picture here. So this is how the aggregation of platelet happens on the subendothelial collagen. Here is a injured tissue. When the platelets, like resting platelets, come in contact with the collagen matrix, then it rolls over, first attached with the BWF and rolls over, and subsequently secretes some prothrombotic molecules like ADP, thromboxane A2, and thrombin, and they attach with each other through fibrinogen and makes a platelet aggregate. And subsequently that increases, and all other blood cells also take part in that clot formation, and subsequently that blood vessel stops, like occludes, and stops blood flowing beyond that point. And so nowadays there are so many drugs available in the market to inhibit like thrombosis, such as aspirin, clopidogrel, and abciximab, and all other drugs, which targets mainly the different signaling proteins in the platelet, or the receptors on the platelets, and or the like any protein in the coagulation system. But all the drugs has one side effect of bleeding, because the balance between inhibiting the thrombosis and restrict the bleeding is very crucial. So our lab is focusing on some targets other than the platelet receptor signaling proteins or coagulation factors, which includes different metabolic pathways and proteins. So out of them, I will present some data which we have recently published on one molecule. So that is, I don't know why it's not moving sometimes.
साइड मूव नहीं कर रहा है.
Yeah, now it's moving. So today I'm going to present some data on role of glycolytic enzyme PKM2, pyruvate kinase M2 in platelet function and arterial thrombosis. As you can see, like pyruvate kinase M2 is a enzyme that is responsible to convert PEP, phosphoenol pyruvate, to pyruvate in the last step of the glycolysis, and it It presents in two forms. One is tetrameric PKM2 and other is dimeric PKM2. And the tetrameric PKM2 helps pyruvate to take that pyruvate towards the mitochondrial cycle, like oxidative phosphorylation. And the dimeric PKM2 favors always the aerobic glycolysis or lactic acid cycle. As you can see, that in resting platelets, both of the OXPHOS and aerobic glycolysis happens, but in activated platelets, so predominantly the aerobic glycolysis happens more compared to OXPHOS. So our hypothesis was to targeting this dimeric PKM2 and down-regulate the aerobic glycolysis, subsequently inhibit the platelet function and arterial thrombosis. So
रजिस्ट्रेशन नंबर नहीं है डॉक्टर का, तो कोई भी कर सकता है, ठीक है? नहीं वो ठीक है, लेकिन रेफरेंस नंबर नहीं है। और बाहर वाले को बिना ग्रुप में आए, मैम के
आपको कर देंगे तो फिर ये नहीं।
yeah, somebody.
No. Now we have muted everybody.
Okay. So although we know that aerobic glycolysis yields a less number of ATP, but we know that a cell always opt for the aerobic glycolysis whenever it needs a lot of energy. So it's known that aerobic glycolysis runs 100 times faster than OXPHOS. So a cell opts for the aerobic glycolysis whenever it needs a lot of energy. So the activated platelets, they need a lot of energy immediately to form a clot formation, form a clot. So it asks for the aerobic glycolysis more. So we wanted to inhibit this dimeric PKM2 and downregulate this aerobic glycolysis, subsequently platelet function and thrombosis. So we used two approaches. One is pharmacological approach by inhibiting PKM2 dimers with a small molecule Ml265, and another is a platelet-specific PKM2 knockout mice in which only platelet has no PKM2, but all other cells and has PKM2. So first, we checked the expression of PKM2 in both human and mouse platelets by the reducing gel. As you can see, sufficient amount of PKM2 is present in human and mouse platelets. As I have already told you, there are two forms of PKM2 present. One is tetramer and another is dimer. So what I've done, like this is a native gel I have drawn to see the two forms of the PKM2 using the same antibody. So as you can see, by activating the platelets with the convoxin or thrombin, you can see these are the dimers. These are increased in the activated platelets, and we successfully inhibited the dimers to some extent by using the two different concentration of Ml265. So this is the quantitative data. As you can see, both the activated platelets without Ml265 has more dimers compared to resting platelets, and in both the concentration of Ml265, we have successfully inhibited the PKM2 dimers. So we first check the agonist-induced platelet aggregation in response to different dose of Ml265 in human platelets. So you might have seen how the platelet aggregation happens. I'll not repeat. In short, we take PRP by centrifuging the whole blood, and we use some agonists like collagen, convulsin, TRAP, ADP, and all other agonists also there to aggregate them. So as the light transmittance increases, then that aggregation of the platelets increases. So as you can see in Ml265, both the doses, like dose dependently, it inhibits the platelet aggregation in response to almost all the agonists. And after that, we check the effect of Ml265 on alpha-2b beta-3 activation and P-selectin exposure. As I have already told you, like after inside-out signaling, after coming with a contact of a stimuli, platelet opens up its alpha-2b beta-3 integrins to bind to the fibrinogen to make a platelet aggregates. So we have stand with PAC1 antibodies with the activated platelets and Ml265 pre-treated platelets in different doses. As you can see, so for these two, like PAC1 and CD62 P binding, I have used this flow cytometry. I think at some point you will learn how to do flow cytometry in this workshop. So as you can see in the both the doses, it decreases the PAC1 binding and as well as the p-selectin binding to the activated platelets in the presence of different doses of Ml265. And also, this is the alpha granule secretion, and this is the dense granule secretion. And this experiment has been done with using the same aggregometer, and I have used a like luminescence substance to see the ATP secretion. So in both the doses of Ml265, we saw a decrease in ATP secretion in response to both convulsine and thrombin. Then we asked whether Ml265 inhibits platelet aggregation in whole blood under arterial shear. So we performed in vitro studies using microfluidic flow chamber assay coated with collagen. So this instrument it's a very good instrument so that you can use the whole blood and label some blood cells to see how the the thrombus formation happens in vitro, like outside the body. It's very good to test the human samples, because in mice we can use them as a live organism to see the thrombus formation. But in humans, it's a great advantage to use this instrument by using the arterial shear, like to mimic exactly, not exactly, but the mimic the uh, flow rate and all the minimal environment we can create, like the subendothelium collagen and also whole blood running over on the top of that at the arterial shear. So this is a great advantage to use this machine for human whole blood using different drugs to test whether it has a antiplatelet or antithrombotic activity or not. So this is how this is just a small video clip. This is a like collagen coated microfluidic chamber, and we run the whole blood and we have labeled the platelets with calcine green. As you can see, like the upper one is a control and the lower chamber is a drug treated one. As you can see, like when the time passes, the accumulation of like platelets and all other cells more in the control compared to the drug here. And this is how we got our result. As you can see, after 5 minutes of flow, the coverage of thrombus is more in the vehicle treated whole blood compared to the ML265. And the percentage of area covered by the platelets also high in vehicle treated platelets compared to the ML265 treated platelets. Based on this compelling data with the dimeric PKM2 inhibitor, we generated a platelet-specific PKM2 deficient mice, and successful deletion of PKM2 was confirmed by the Western blotting. And next, we checked the platelet morphology and ultrastructure in the absence of PKM2, and TEM confirmed the normal platelet morphology. Then we determined the effect of PKM2 deletion on platelet aggregation. As you can see, like we did not find so much inhibition in the higher concentration of agonist, but at the suboptimal doses of different agonists like convulsine, collagen, PAR4, thrombin, and ADP, we found a drastic inhibition of platelet aggregation in PKM2 flux PF4 cray mice, that is platelet specific PKM2 knockout mice. Then we checked the clot retraction capacity in PKM2 deficient platelets, and we found that lack of PKM2 in the platelets makes them less retractable, suggesting that the outside in signaling is also affected in the absence of PKM2. Next, also we checked the aggregation capacity in whole blood in PKM2 platelet specific PKM2 knockout mice, and you can see using the dynamic flow chamber assay, as you can see in PKM2 specific knockout mice, we found a less thrombus formation compared to the control mouse. and the percentage of area covered by the platelets were also less. Okay, so this is the most advanced technique nowadays to see the thrombus formation in vivo. So next we performed ferric chloride injury induced carotid thrombosis assay to confirm the in vitro data in vivo. So what we do in this model, you can learn at some point of in the workshop. So we anesthetize the mice and we exercise the sometimes mesenteric artery and carotid arteries and expose them with some percentage of ferric chloride to make an injury on top of that. And then we see that live thrombus formation in a anesthetized mice. It happens up to maybe 1 hour, and we see the, as you can see in a like video clip, how the thrombus formation happens after a ferric chloride injury. So you can get a lot of data from this to analyze how the thrombus formation happens, like what is the rate of thrombus formation. And as you can see, like this is a PKM to flux means the control mice. The white lines are delineated by the vessel, the arteries, and in different time points, you can see in the control mice, the thrombus increases, but compared to the control mice, in the knockout mice, you can see the decrease in thrombus formation here. And also we note the time of occlusion. So at what time the flow of that blood vessel completely stopped. So that you can see here the time to occlusion was around 15 to 17 minutes in control mice, and that was extended in knockout mice. And the rate of thrombus growth was also decreased in knockout mouse. After getting this data, we are thinking that the bleeding time will increase in the knockout mouse, but we didn't like. It's interesting not to get any difference in bleeding time in both the mice. So that's really fascinating because this ML265 can be explored as an antithrombotic drug because it is not having any effect on bleeding time. Like it is affecting thrombosis, it's inhibiting thrombosis without affecting the hemostasis. So that was in the male mice, and to test whether this effect is not only sex specific, we performed the intravital thrombosis experiment using the female mice, and also we found the longer occlusion time in female mice and less thrombus growth in female mice, and also we got like no difference in bleeding time. So to test that whether this effect is not only the model specific, so I have used like another model using the laser injury induced mesenteric artery thrombosis model, and I can show you through a video clip how I use the thrombus to make an injury in the artery, in the mesenteric artery. So here you can see these are the laser bombardments you can see here. So then, yeah, then you can see how the fluorescently labeled platelets are attaching to the site of injury during course of time. It happens through maybe 2 to 3 minutes or 1 to 2 minutes and then diminished. It's a long video, so I don't want to play it all. But this is a different different model of thrombosis other than ferric chloride. So we can make the thrombus in smaller vessels like mesenteric artery and also cremaster muscles. So we can use different small arteries for this model. So by using this model, we also got a less thrombus formation in knockout mice compared to control mice. And this is the fluorescent intensity we can measure, and we can measure the area under curve, whether the fluorescently labeled platelets are more in control or in the knockout mice. As you can see, the control mice is bigger thrombus compared to the knockout mice. It's not moving again. I think it will take some time.
Sir, please tap on the PPT so that it will move. Yeah.
Yeah. So then, after confirming the role of PKM2 in modulating thrombosis in vivo, we sought to determine the inhibitory effects of Ml265 on thrombosis in mice. So the time to complete occlusion increased in both the dosage of Ml265 when we infused into the mice, wild-type mice. As you can see, the thrombus growth was also retarded a lot in inhibited infused mice. And most interesting thing is that like it is also not affecting hemostasis. So we are proposing ML265 as a antithrombotic drug which will inhibit efficiently the thrombosis but not affect the bleeding time, or it will not occur any bleeding complications. And previous reports already showed that, to go into a little bit deeper into the mechanism, showed that PKM2 promotes cell migration and inhibits autophagy by mediating PI3 kinase AKT signaling pathways. That contributes to the development of some cancer. So PI3 AKT pathway is also known to be involved in the inside-out signaling and outside-in signaling during platelet activation. So because the pretreatment of human platelets with ML265 downregulated both inside-out signaling and outside-in signaling in stimulated platelets, so we further determined whether this targeting PKM2 with ML265 attenuates the PI3 kinase AKT pathway. As you can see in human platelets, it decreases the phosphorylation level of both AKT and GSK3. And that also we tested in the knockout mice, that was decreased in both the convulsine and thrombin treated platelets. These are the quantitative data on the right hand side. So in summary, we found that like platelet activation is associated with the elevation of PKM2 dimer formation, and it modulates multiple aspects of platelet activation and thrombose formation in vitro. And platelet-specific deletion of PKM2 inhibits the platelet activation and thrombosis in vivo as well. And targeting PKM2 inhibits arterial thrombosis without altering hemostasis in vivo, and PKM2 regulates PI3 kinase mediated AKT GSK3 signaling. So this is the end of this project. I want to give a glimpse to the deep vein thrombosis too. As you all know, deep vein thrombosis is a threat to all the mankind now, and it happens in the deep vein, and it can loosen up and travel through the circulation and can form pulmonary embolism. So I'm also doing some deep vein thrombosis models. It's not moving.
Tap it on, sir. Tap on the slide.
Yeah. So this is a stenosis model I have done. In stenosis, what happens, like I ligate the deep vein from deep inferior vena cava, and ligate the side branches and put a needle spacer here and take it out after the ligation to make the flow around 20% and 80% blockage. And we confirmed it with the ultrasonography in the mice. As you can see, there is a space over here, and this is the thrombose developed here, and we are trying to measure this thrombus by the ultrasound, so we can follow this thrombus during the course of time to see the thrombus resolution. So, and this is how thrombus form in the inferior vena cava. So we can measure the thrombus length and the thrombus weight as well. These are the 12 weeks C57 block 6 mice from the Jackson laboratory. So at last, I want to acknowledge Professor Anil Chauhan, my mentor, and Dr. Stephen Lanes, Sanjana Dayal, and Janish Tabor in our group, and our collaborators, Matthew Pothop and Massimo, and the UIOA core facility of microscopy, flow cytometry, and metabolic core. And I want to really thank to American Heart Association for funding me during the course of my research. So thank you so much. Now I'm open for any question.
Thank you, Manas. Before we go for questions, just a word to the audiences so that they can organize their thoughts in this. So Manas' work is a example that how biochemical pathway can be extrapolated to thrombosis. So you see that those who are from the biochemistry department, they can apply their own knowledge and stress their imagination to find some of the solutions or mechanistic basis for this thrombotic process. So this is a very new concept that PKM2 is having this inhibition of PKM2 having a good effect. And as I told you yesterday also, PKM2 inhibition has been tried as an anti-cancer effect also. So don't forget what I told yesterday. There is a lot of similarities between the cancer cells and platelets, except that one causes cancer and other does not cause cancer. That is the only difference. But the rest of the thing, you can put anything that already well known in cancer studies and try to explore with these tools in the platelets. I think you can get some novel gateway. Thank you, and open for questions.
Unmute. Now everybody has been unmuted. Please ask your question if you have any.
Very good morning, Manas sir. I'm Shwetanka Prasad from Manipal Tata Medical College, sir. Sir, for seeing this platelet activation in the cancer cells, according to you, which one is the best sample, sir? Best sample means either it's from mice or from human beings, sir.
Can you repeat 1 second your question?
Means what is the sample of choice, sir? Means blood sample for seeing the activation of platelet in cancer. We have to see the blood samples from human beings or from mice.
Yeah, like it's a great question. So you were asking about cancer-associated thrombosis, right?
Yes, sir.
So, okay. So to see the cancer-associated thrombosis, you can obviously you have to take the human blood samples to see the extent of platelet activation. Like I have told in my presentation, like you can check the PAC1 binding by staining the PAC1 antibodies with the human platelets, and also you can check P-selectin exposure by taking the human samples. But by using different cancer mice models, you can check different drugs also. So because nowadays cancer-associated thrombosis is emerging a great opportunity compared to only thrombosis and only cancer. So now we can make some different like cancer mice models and do some perichloride or laser injury models to see how we can combat that, inhibit that thrombosis, like which is cancer associated. So nowadays, as sir told, like both the cancer cells and platelets, activated platelets, they behave very similarly because both of the cells, they need a lot of energy in a very short span of period. So we can just see the signaling pathways, how they interact, and many factors like platelets secrete to the surrounding that helps the cancer cells also migrate, like the metastasis also occurs by the activation of the platelets. So they work hand in hand. So when we can inhibit the platelet activation or thrombosis, subsequently we can also inhibit the cancer metastasis also. So this is a great question, and we can use both the human blood and platelets to see the extent of platelet activation in cancer patients, and also we can use the different cancer mice models to test different drugs. Thank you so much for this question.
Thank you so much, sir. Thank you.
Hello, sir.
Yes.
Yes, good morning, sir. Very nice talk. I have
Thank
a
you.
question. As you mentioned, like deficiency of the platelets and all, can I know how do you, can I the same approach, is it possible by VVW factor, Van Wille Brand factor deficiency kind of thing?
Sorry, can you repeat? It was cutting
Maybe,
in between.
so wherever you mentioned like platelet deficiency model and all, creation of that. So
Okay.
what is the further consequences in the mice model? Just I want to know that.
Still, I don't get your question. Could you please just
So,
simplify?
as you mentioned that,
Yeah.
platelet deficiency model in mice.
Like I explained about the PKM2 deficient platelet specific PKM2 deficient model. So we can knock out the single gene, the gene of interest, in only platelets. If you want to delete it from any kind of cells, like I have used PKM2 flux PF4 cray mice. So according to your cells of choice, if you want to delete some of the protein, maybe BWF, any other protein from any type of cells, maybe you want to delete it from neutrophils. So there are some or endothelial cells or myeloid cells. Like let's say if you want to delete them from myeloid cells, so you can just cross them with lysine cray. So it's a specific receptors only present in the myeloid specific cells. So obviously you can make different cell specific and tissue specific knockout models to use your
Yes,
experiment.
so how long the mice will be survive after remove this deletion? That's I want to know.
They are fine. They are fine with the
They are
deletion
fine.
of with the deletion of PKM2 from platelets. They are absolutely good. And we have checked the weight, we have checked the blood, all other blood platelet and all other blood cells counts. So they are quite good.
Okay. And one more thing, sir. And as you mentioned that, is there any kind of studies like platelet mother cell and megakaryocyte? If you remove that same gene in megakaryocyte, whether it will convert to the platelet or it will be deplete of. That's I want to know. Yes.
The PF4 cray mice I have used, that's only for megakaryocytes. So that is taking out from the megakaryocytes, as the platelets are coming from the megakaryocytes, they are also devoid of PKM2 as megakaryocytes. So they are coming from megakaryocytes. You are right.
Yes, sir. Okay. Thank you so much. Yes.
No problem. You're welcome.
Good morning, sir.
Yes.
Sir, it's Gaurahari Sahu from Central University of Punjab. Sir, I want to ask a question. Previously, I have read one paper on that glycolysis elevates the platelet activation. There is a mention of DASA that activates PKM2, right? And PKM2 blocks the GLUT3 pathway. The GLUT3 channels PKM2 blocks that. But we also know that glycolysis is necessary for the platelet activation. So if PKM2 is blocking the GLUT3 channel, then how PKM2 as an enzyme is helping in the activation of platelets in the glycolysis context?
Yeah, okay. So it's a very good question. Actually, PKM2 exists in two forms, as I told you. Like one is tetrameric form and other is dimeric form. The DASA you told, that's X like similar to ML265, what I have shown you in the slides. Okay. So you were telling DASA inhibits the GLUT3 pathway. The GLUT3 pathway is responsible to the absorption of glucose inside to the cells, right? So we have also seen the less glucose uptake by using the ML65 also. So you are right. So it inhibits the glucose uptake also. And what is the next question? Second part?
Sir, the next question is that if PKM2 is inhibiting the GLUT3 and the
Not
platelet is
PKM2.
needing,
Is it PKM2 or DASA?
sir,
DASA. Yeah. Because PKM2 exists in two forms, like in tetramer and dimer. Like, you know, PKM1 is only exists in tetrameric form, that only favors towards oxidative phosphorylation, that takes pyruvate from the glycolysis to the mitochondrial oxidative phosphorylation and yields a lot of energy. The PKM1, like PKM1, PKM2 tetramers also helps in oxidative phosphorylation, taking the pyruvates to the mitochondrial cycle. So our motto is to inhibit the dimeric PKM2, which takes the pyruvate towards the lactic acid cycle or the aerobic glycolysis.
okay,
Because
sir.
aerobic glycolysis that happens 100 times faster than oxphos. So at the time of need, platelets extract the energy from aerobic glycolysis compared to oxphos in the early stages. So that's what we are inhibiting, the aerobic glycolysis, by inhibiting the dimeric PKM2 by ML265 and by using the genetic transgenic model by having the platelet specific PKM2 knockout mice. Is it
Sir,
clear now?
yes, sir. Sir, DASA is a inhibitor or activator of PKM2?
Okay, this is a confusion for most of the people. So you can call that a activator of PKM2 tetramers and inhibitor of PKM2 dimers. We call it that PKM2 dimeric inhibitor because we are inhibiting the PKM2 dimers and we are not caring about PKM2 tetramers. But exactly what the inhibitors does, it inhibits the PKM2 dimers but activates the PKM2 tetramers. That's the really function of the PKM2, pyruvate muscle kinase M2, that really helps the pyruvate to take towards the oxidative phosphorylation. So that is a activator of PKM2. In some paper it has been written. So activator means it's activating the PKM2 tetramers. And when I'm telling the inhibition of PKM2, that is the PKM2 dimers, inhibiting the PKM2 dimers. They tell exactly the same thing.
Okay, sir.
Yes, sir.
Thank you, sir.
I have one more question, sir.
Mm-hmm.
Yeah, as we mentioned that there is a similarities between cancer cell and platelet, and I have a doubt. So since the platelet is the anucleate cells, and moreover the life is very limited, and whereas the coming to the cancer cell that is immortal in nature, moreover it is a nucleated cell. So how you can
Yes.
compare with the metabolism and all? Just I want that's.
Yeah, that's a great question
Yes.
also. Like,
Yeah.
you are right. It's a nucleated cell and this is a anucleated cell. But
Yes.
the need of the, like, both the cells, both cancer cell and activated platelets are same. And moreover,
Yeah.
they share like many signaling pathways, very similar signaling pathways. Because a cancer cell needs a lot of energy, you know, like it absorbs a lot of glucose and also it shut down its mitochondria and runs aerobic glycolysis for the immediate need of energy, the cancer cells. Likewise,
Okay.
platelets, when they comes in contact with different type of stimuli like subendothelial collagen or ADP or thrombin, any type of stimuli, so they acts like a cancer cell. They need immediate need of energy, and they
Yes.
to carry forward that in energy consuming process of clot formation and thrombus formation. So I will not say they are exactly doing the same thing, but most of the things are similar in between activated platelets and cancer cell.
Yes. Okay. Thank you so much. Yeah.
No problem. You're welcome.
Sir, one more questions from my side.
Yes.
Sir, in between DASA and the ML265, in DASA we have seen that the bleeding time is more in case of tail bleeding assay. And as you have mentioned that in case of ML265, it's not affecting the in vivo hemostasis. So is this the only benefit of ML265 over DASA? Because DASA also affects, also inhibits the PPP pathway. So is like ML265 is also inhibiting the PPP, or only just because of hemostasis issue we are taking ML265 is more superior than DSA?
No, I'm not saying about like ML265 is superior or DASA. And actually, I've not checked the effect of DASA in thrombosis and hemostasis. I cannot comment on that. But as long as ML265 is concerned, we have checked the tail bleeding time, and it is not changing the tail bleeding time whenever like we have used the high dose of like 50 mg per kg. But I have not checked the DASA effect on bleeding, tail bleeding in mice.
Yes, thank you.
No problem.
So,
One last question, sir, from me. Shwetanka Prasad from Manipal Tata Medical College, sir. Sir,
Yes.
Is there any association or correlation between activated platelet and basophil, sir?
B.H.U, I'm not aware of
In
that.
cancer cells, sir. Means,
Yeah,
is
I'm
there
not.
any association or correlation between activated platelet and basophil in cancer cells, sir?
Yeah, I have a great interest to work in like cancer-associated thrombosis, but I have not worked on that till now. But yeah, it's my future goal. So I'm not aware of that actually. Sorry.
Platelet neutrophil interaction. The next speaker is eminent speaker, Professor Kemparaju. He can, at the end of his talk, he can little bit speak about platelet neutrophil interaction. So he's waiting for the.
Thank you, sir. Thank you, Manas, sir. Thank you, sir.
Thank you.
I thank Manas sir for his elaboratory talk and also sharing with us the latest research that he's been doing. And also I thank for his patient listening and answering most of the queries of the participants. And despite of the time zone differences, he has managed to come in time and make this event a very nice one for us. Thank you again. And now I'll invite Professor Kemparaju sir. I have been able to listen him in person when he has visited our department a few years back before COVID. And sir is a professor in Department of Biochemistry in University of Mysore, and he has been doing his very novel research, which includes snake venom toxins on different type of platelet functions. And also he has very very good publication in Nature Communication, in Journal of Pineal Sciences, Hematologica, and Free Radical Biology Medicine and ACS Chemical Biology. So, being despite being in a state university, he has achieved that most of many of us cannot, and he has been a very he has done very legendary work in that area. And he has many good publication which with H index of almost 42 and IT index of 89. He has received many awards and accords, and he has guided 18 PhD students. He had done many projects with DST, SERB, UGC, DBT, direct, and he has been external reviewer in many important and prestigious journals, including Lancet, the British Journal of Pharmacology, Current Medicinal Chemistry, Current Topics in Medicinal Chemistry, and many others. And he has been a life member of SBC India, Indian Science Congress, and Indian Society for Atherosclerosis Research. I welcome again Dr. Keparaju sir for his elaboratory talk. Now we'll be having his screen shared so that he can start his presentation. Thank you, sir.
Am I audible?
Yes, sir. Yes, you are very audible.
Thank you so much for a nice introduction about me. It's an elaborate introduction anyway. Thank you so much. Thanks a lot. Good morning, all of you. First of all, I'd like to extend my sincere and heartfelt gratitude to Dash sir. To summarize my relationship with Dash sir, he is my mentor, he is my model professor, model guru. I always seek his blessings. Thank you very much, Dash sir. Thanks a lot for you. I also extend my sincere thanks to Nitesh for all communications done. You already mentioned I am from a traditional university, University of Mysore. I am from a setup where sometimes we struggle for distilled water, and the high-end technology that Manas and I discussed so far, it's a dream for us. Manas and I actually, yeah, it's a wonderful talk. I really enjoyed it, but I just joined a little late. I could not grasp the initial things. So it was a wonderful talk. It's really good. I really congratulate you. Uh, uh, you know, it's not sharing.
Now your slide is visible, sir. Try
Yes,
it on the
sir.
slide.
It's visible now.
Yeah.
Now it's visible.
Yes, sir. Thank you, sir.
Yes, sir. Our group at the Department of Biochemistry, University of Mysore, is working on different concepts, different facets of snake venom pharmacodynamics. Basically, I work on snake venom toxins. So my group is currently focusing on the different areas like mechanism of viper bites induced sustained tissue destruction at the bite site. You can see over here. This is the major thing that we are pursuing on right now. And venom-induced oxidative stress and hypoxia, venom neutralization strategies by antivenom and small molecules, characterization of molecules of therapeutic importance from venoms, and effect of venoms or venom toxins on hemostasis. So to fall in line with the interest of the scope of the current workshop, my current talk is focused on the last concept, effect of venoms or toxins on hemostasis, particularly plasma coagulation and platelet aggregation. So as a result, this is the title of the topic I'm going to talk about maybe for the next 35 to 40 minutes. Anticoagulant and antiplatelet properties of a metalloprotease from Indian cobra naja venom. When I say hemostasis, it means a controlled, a tightly regulated crosstalk between blood plasma and the platelets in the first line defense, particularly in innate immunity and eventually in wound healing process. When I say snake venom is a complex mixture of predominantly enzymatic and non-enzymatic protein and peptide. Incredibly, most of the toxins are target specific. They do target vital organs, vital systems such as nervous, muscular, and circulatory systems. These three systems are tightly interconnected, interlinked, such that effect on one system invariably affect the other two, resulting in immobilization or killing of the prey animal or human victims. This is the SDS page pattern of the Indian saw-scaled viper venom. You can see lot many protein bands, protein toxins resolved in the SDS page. If you take the same venom and carry out the RPHPLC, you get as many as 100 different peaks, suggesting that snake venom is a complex mixture of protein and peptide toxins. So thus, many venoms and venom toxins directly affect the circulatory system. That means affect the hemostatic system. In that, particularly plasma coagulation and platelet aggregation events. If you take plasma coagulation, so many toxins, they promote coagulation of plasma or coagulation of blood. They are called procoagulant toxins. Many toxins, they inhibit coagulation. They are called anticoagulants or anticoagulant toxins. And many toxins interfere in platelet function. They induce aggregation, and many would inhibit aggregation of human platelets. So if you take hemostasis is an acute phase response to vascular injury. It is characterized by platelet activation, clot formation, and clot dissolution. Whenever there is vascular injury, immediately platelets get activated. They bind to the damaged site. Right on top, there will be formation of clot, which seals of the damaged site temporarily. So thereby, day-to-day minor hemorrhages are taken care of. So whenever there's it's something like platelets act like bricks over which we have the cement, the clot, a temporary wall is created. It's something like that. So if you take the blood coagulation event, we have intrinsic pathway. It is also called contact activation pathway. Extrinsic pathway, it's also called tissue factor pathway. They both get activated through different mechanisms, but they culminate at one point, at the point of formation of factor 10 to 10A. So from 10A onwards, we have the common pathway. So common pathway results in the activation of the prothrombin to thrombin. Thrombin again act on fibrinogen, convert fibrinogen into fibrin. That's the clot. So the factor underscore in this event is that each factor activates the other factor through limited proteolysis. As a result, there will be a formation of a cascade of events. And another important thing that you come across in this event is that the property of fibrinogen. The soluble fibrinogen is converted into insoluble, the fibrin clot, when it is subjected for limited proteolysis. So the efficiency of final clot formation depends on the efficiency of all the clotting factors. If there is an impairment in any of the clotting factors, resulting in impaired the clot formation, leading to bleeding disorders. Thus, toxins, snake venom toxins, strongly interfere in coagulation event. Many toxins affect the intrinsic pathway. Many other toxins affect the extrinsic pathway. Many will work through even common pathway. So this is just to give you, this cartoon is giving you the formation of platelets. Platelets are anucleate biconvex discoid cells derived from the multinucleate giant cells of the bone marrow, the megakaryocytes. See, platelets are formed from the bone marrow. However, recently, in 2017, a group from California University, they have demonstrated the formation of platelets even from lungs. Nearly 50% of the platelets that are present in our system are generated from the lungs as well. So in the lungs, megakaryocytes sits, and from the megakaryocytes, platelets are formed. But how exactly megakaryocytes transferred from or move from bone marrow and settle in lungs is not been clearly understood. This is just to give you the gist of the general mechanism of platelet aggregation. I hope many of the speakers might have already discussed about this, but never mind. I'll go through this quickly. So platelets in the resting stage, that is in the unstimulated state, they are they are like this biconvex discoid in shape. But when the platelets stimulated, they are activated by agonists. We have various agonists here. We have shear stress, epinephrine, collagen, thrombin, ADP, thromboxane A2, arachidonic acid, platelet activating factor, histocitin, antigen antibody complex, calcium ionophores, and many more. They serve as agonists and activate platelets. When platelets get activated, so a lot many things will occur. Number one, platelets become sticky. Number two, platelets undergo degranulation. And number three, platelets change their shape like this. So the stickiness, degranulation, shape change, nearly they occur almost simultaneously. See here, when the platelets are in the resting state, they are non-sticky, they are like biconvex as I said, and when they are activated, the final effector receptor, the major receptor is the glycoprotein 2B3A complex. It is also called alpha 2B beta 3, and it is popularly referred as fibrinogen receptor. This receptor is actually exhibit broader specificity. It not only binds fibrinogen, but it also binds other ligands as well. So like fibrinogen, van Willebrand factor, fibrin, fibronectin, mitronectin, thrombospondin, and so many others. So it exhibits broader specificity, but the predominant ligand would be the fibrinogen. So that's why this receptor is popularly called fibrinogen receptor. In the resting platelets, the fibrinogen receptor actually remains in a conformation which is inactive. It cannot bind the ligands. But when the platelets are activated, it undergoes conformational change such that it can now bind the ligands. So the ligands here, as I said, fibrinogen, one membrane factor, fibrin, fibronectin, vitronectin, thrombospondin, these are the major ligands. They cross-link the platelets. See, the interesting thing in this case is that all these ligands interact, they bind the receptor, activated receptor, through a specific protein motif. They have a motif, specific motif called, the motif has RGD sequence or KGD sequence. RGD means arginine, glycine, and D aspartic acid, or K lysine, G glycine, D aspartic acid sequence. Sorry. Oh. Aspartic acid sequence. So this is what? This is a common mechanism. Whichever the agonist stimulate the platelet, the end result is almost the same. So they activate the glycoprotein 2B3A complex through inside-out signaling, and there's a degranulation. The granular materials are secreted. Most of them are ligands here. So eventually, the platelets are cross-linked. So in degranulation, if you take into account, platelets are loaded with three types of granules: alpha granules, dense granules, and lysosomes. Alpha granules have many number of molecules, many number of different types of substances, say fibrinogen, one membrane factor, fibrin, fibronectin, all these. So something like 30 to 40 different components are stored in alpha granules. And so also in dense granules, ATP, ADP, GDP, magnesium, serotonin, phosphates, chemokines, factor 13, and alpha-2 macroglobulin, platelet factor 4, and so many other ligands, other struct components are stored in dense granules. And lysosomes. Pardon? The problem with the network? Oh, I'm okay. I'm okay from my side, but
ये तो सेम तो वही लगा है.
hello. Hello. Is it okay now?
इसमें सुनाई दे रहा है क्या?
Is it okay? Hello. Oh.
जिनके पास ईयर
Oh, it's voice is breaking. Voice is breaking from your side.
उनको सब वो नहीं थे
Oh, it's breaking.
करने
Is it okay now? Is it okay now? Hello. ಏನು ಪ್ರಾಬ್ಲಮ್ ಬಂತು? ಪ್ರಾಬ್ಲಮ್ ಅವ್ರ ಪ್ರಾಬ್ಲಮ್ ಸ್ವಲ್ಪ ವೇಟ್ ಮಾಡ್ತೀರಿ ಇಲ್ಲೇ ಇರು ಹೊರಗಡೆ ಬೇಕಾದ್ರೆ Nitesh. Oh, voice is not a problem. What about slides? Slides? It's working. Okay, okay. I'll continue. Okay. Okay, I'll link. So whichever could be the agonist, the end result is the same. So there is in signal transduction, inside out signaling. This fibrinogen receptor gets activated that's under a conformational change. So the active receptor now binds the ligands. The ligands will intercalate the platelets. So eventually platelet aggregation takes place. So here the most important thing is ligands binds through a specific structural motif that contains RGD or KGD sequence. Okay, so now as I already mentioned, snake venom is a complex mixture of enzymatic and non-enzymatic protein and peptide toxins. So we have variety of enzymes have been isolated and characterized extensively from different snake venoms. So we have phospholipases A2 proteases, so represent a family of proteases over here. We have acetylcholinesterases, heparinases, hyaluronases, L-amino acid oxidases, deoxyribonucleases, and so many enzymes here. And if we come to non-enzymatic toxins, we have three-finger toxins. Three-finger toxins include cardiotoxins as well as neurotoxins. And disintegrants. Disintegrants, these are the small molecular weight peptides, the molecular mass ranging from 5 kilodalton to 7 kilodaltons. These are basically inhibit platelet aggregation. So disintegrants, they inhibit platelet aggregation because they possess RGD or KGD sequences. That this you have to remember. And CTLs, that's C-type lectin-like molecules, protease inhibitors. We have Cunitz type of protease inhibitors, BPPs, bradykinin potentiating peptides, NGF nerve growth factors, ANP natriuretic peptides. These are all non-enzymatic toxins. Thus, so snake venom is a mixture of enzymatic and non-enzymatic protein and peptide toxins. These are the various pharmacological properties exhibited by snake venom poisoning. So we have edema, hemorrhage, neurotoxicity, myotoxicity, cardiotoxicity, hemolytic activity, coagulant activity, platelet aggregation, hypertensive, methemoglobinemia and hypoxia, netosis and etosis. These last two, methemoglobinemia and hypoxia and netosis or etosis, were discovered from our laboratory. So when I say coagulant, as I already mentioned, some of the toxins, they promote coagulation. They are called procoagulants, and some will inhibit coagulation. They are anticoagulants. So coagulant activity could be procoagulant activity or anticoagulant. And platelet aggregation also. Some of the toxins induce aggregation, and some would inhibit aggregation. So thus, snake venoms interfere strongly in hemostasis. Yeah. I continue. Shall I continue?
Yes, sir. Yes, sir.
Yes. This is to summarize the properties of a purified metalloprotease from Indian spectacled cobra naja venom. This is the SDS page pattern. You can see over here a sharp single band suggesting its purity. Its purity was further confirmed by MALDI-TOF mass spectrometry that revealed a single peak with the molecular mass of 67.80 kilodalton. The proteolytic activity was inhibited both EDTA and EGTA, suggesting it's a metalloprotease. And atomic absorption spectrometry confirmed the presence of zinc and calcium. And now if you take the mole to mole ratio, 1 mole protein to 1 mole zinc to 1 mole calcium, that is the ratio they form a complex. And when we check the various toxic properties, our protease is devoid of all these toxic properties. So it is devoid of edema, devoid of hemorrhage, neurotoxicity, cardiotoxicity, myotoxicity, cytotoxicity, and lethality. So thus it is non-toxic in nature. However, our protease strongly interfered in hemostasis. It was exhibiting strong anticoagulant activity, that is inhibited the plasma coagulation and also inhibited platelet aggregation. Thus, strongly it inhibited hemostatic function. So this is to show the plasma recalcification time. We have used citrated human plasma. So this is the dose dependent effect. This is the time dependent effect. And this is the control here. so control, and in presence of our protease, we can see increasing the clotting time. So here also, it's a time-dependent effect. There is increase in the clotting time, suggesting that our protease showing anticoagulant activity. It is inhibiting the formation of the clot. So since it is inhibiting the formation of clot, we wanted to understand the mechanism of anticoagulant activity. So in order to get the insight to the mechanism of action of anticoagulant activity, so we tested APTT, PT, and TCT. APTT means activated partial thromboplastin time. This works through intrinsic pathway, and PT, prothrombin time, it works through extrinsic pathway, and TCT, it works through a common pathway. In all these cases, we observed a delay in clotting time, suggesting that our protease is not working through intrinsic pathway or extrinsic pathway or common pathway. It is not working through these. So that anticoagulant activity is not through these pathways. That means the remaining things are probably it is interfering in fibrinogen function and fibrin clot as well. So thus is the anticoagulant activity exhibited by our protease is due to degradation of fibrinogen, that is clotable fibrinogen, or degradation of fibrin clot itself, or degradation of both, that is clotable fibrinogen and the formed clot. As a result, anticoagulant activity. So for that reason, we check the fibrinogenolytic activity of our protease. This is the fibrinogenolytic activity, dose-dependent effect here. This is the control fibrinogen. We have A alpha, B beta, and gamma chain. So this is resolved in SDS page under reduced condition. So in presence of our protease, you can see the disappearance of A alpha and also B beta chain here. So our protease cleave A alpha chain in preference over B beta chain, while gamma chain remained resistant. So here you can see over here, gamma chain remained resistant. So it is not acted upon by our enzyme. So it's only A alpha and B beta chains are cleaved. This is the dose dependent effect. This is the time dependent effect. So we also check the fibrinogenolytic activity on plasma proteins. So whether our protease is acting on any other plasma proteins other than fibrinogen also, that we check with this. So this is the SDS page pattern of the plasma proteins over here. This is the control one. You can see over here the top one, the second band is the fibrinogen band. You can see over here, specifically fibrinogen band is cleaved, suggesting that our protein specifically is cleaving fibrinogen. Fibrinogen, so our enzyme specifically acting on fibrinogen, it's a fibrinogenolytic enzyme. So thus the anticoagulant activity is due to the depletion of clotable fibrinogen. So is the anticoagulant activity could also be due to the depletion of fibrin clot. So this is what. So for that, we checked the fibrinolytic activity. We used the fibrin clot and tested the effect of enzyme on fibrin clot. So fibrin clot, this is the control. We have alpha polymer, gamma gamma dimer, alpha chain, and beta chain here. See, you can see over here, our protease selectively degraded alpha polymer and alpha chain. It did not degrade gamma gamma dimer and beta chain. It degraded only alpha polymer and alpha chain. Okay, this is the dose dependent effect. This is the time dependent effect. We used washed plasma clot for the experiment. So this is again the same fibrinolytic activity in a different method. So we used a plate method over here. What we did was we took the plasma and molten agar, so mixed the things uniformly, and we initiated clotting in the plate using calcium chloride, and we checked the fibrinolytic activity here. This is with increasing doses of our enzyme. A, we have our protease, that's 1 microgram, B 2 microgram, C 3 microgram, so 4 microgram. So there is a dose dependent. As the dose increases, the cleavage one, the translucent side increases now gradually as the dose increases. So E is in presence of PMSF. PMSF is actually serine protease inhibitor. PMSF did not inhibit the enzymatic activity, while EDTA, F-EDTA, inhibited the enzymatic activity. you could see no degradation sign over here. So again, we tested the degradation of whole blood clot as well as plasma clot. So this is the blood clot, whole blood clot, that's dose dependent effect, and this is the plasma clot. So the plasma clot was effectively degraded over whole blood clot. However, both whole blood as well as plasma clots were degraded or hydrolyzed by our protease, cobra-venom protease. Thus, the anticoagulant activity of our enzyme is due to the fibrinogenetic activity as well as due to fibrinolytic activity. It has both fibrinogenetic and fibrinolytic activity. So our enzyme removes the clotable fibrinogen and also degrade the formed clot, so resulting in the anticoagulant activity. So the anticoagulant activity, that's our anti-hemostatic effect, could also be due to the inhibition of platelet aggregation as well. So this general mechanism of platelet aggregation I've already explained, and the most important thing in this case is that I told you the fact that many venom toxins interfere in platelet aggregation events. So thrombin-like enzymes, they induce platelet aggregation generally. Thrombin-like enzymes present in venoms, they induce aggregation. So thrombin-like enzymes, they are proteases anyway. And disintegrants, especially, as I mentioned, disintegrants are small molecular weight peptides of molecular mass ranging from 5 kilodalton to 7 kilodalton. They contain RGD sequence. With the help of RGD sequence, they can compete with the binding of the ligands to the activated receptor here. So we have the ligands here. The ligands cross-link to activated platelets. So the disintegrants, with the help of RGD motif or KGD motif, they can also compete with the ligands, real ligands. So let's say these are the disintegrants I've just mentioned through the cartoons here. So let's say these are the disintegrants. Disintegrants can compete with the real ligands and can inhibit the platelet aggregation. So we have the drugs which are already available in the market, in the US market, Agrestat, Integrelin, FTP-tide. See, all these drugs are made using disintegrants as the lead molecules. So disintegrants contains, as I said, RGD and KGD sequences. And these ligands also contain RGD and KGD sequences. So these ligands bind the platelets through RGD and KGD sequences. So disintegrants will have RGD and KGD sequences. With the help of RGD and KGD sequences, they can compete for the binding to the receptor, and hence, they can inhibit the aggregation, platelet aggregation. So we checked our enzyme whether what agonist-induced aggregation is inhibited. We checked the collagen-induced aggregation, ADP-induced aggregation, epinephrine-induced aggregation, ristocytin, arachidonic acid, thrombin. However, so here you can see over here the collagen-induced aggregation was inhibited 100%, while ADP-induced aggregation was inhibited to the extent of 60 to 70%, while epinephrine-induced aggregation was inhibited again partially about 50 to 60%, while ristocytin or arachidonic acid or thrombin-induced aggregation was not touched. It did not interfere in ristocytin, arachidonic acid, and thrombin-induced aggregation. So it interfered in collagen, ADP, and epinephrine-induced aggregation. So to take it forward, we just restricted ourselves to collagen-induced aggregation for further studies. So this is the collagen-induced aggregation trace. So this is the control aggregation trace of collagen. So in presence of our protease, that's NNPF3, you can see there's a complete abolition of the aggregation of platelets. See, our protease is working at the nanomolar level. 150 nanomolar, at 150 nanomolar is completely inhibiting collagen-induced aggregation. And see here, we also check the platelet aggregation inhibition property, whether it is protease activity dependent or even non-enzymatic leak can inhibit platelet aggregation. So our enzyme inhibiting the aggregation through non-enzymatic mechanism because EDTA is knocking off the enzymatic activity, inhibiting the enzymatic activity. In spite of inhibition of enzymatic activity, there is inhibition of platelet aggregation, while this is a native enzyme. Okay, so the inhibition of platelet aggregation inhibited through both enzymatic as well as non-enzymatic mechanisms. How does the protease inhibits collagen-induced aggregation? Does the protease deplete or mask collagen or collagen receptors? So we have collagen receptors. We have two types: alpha-2 beta-1, which is a minor receptor, and glycoprotein 6, which is a major receptor. So does the protease deplete or mask collagen receptors, alpha-2 beta-1 or glycoprotein 6, or both? Or does it mask or deplete fibrinogen, or does it mask or deplete the fibrinogen receptors, that is alpha-2b beta-3 or glycoprotein 2b 3a complex? So we check the effect of our protease on collagen. You can see over here, this is type 1 collagen, this is type 4 collagen. Our protease did not cleave collagen at all. It did not deplete the collagen. And next, we did the addition assay, so targeting alpha-2 beta-1 integrin. So what we did was, so we have alpha-2 beta-1, it's a minor collagen receptor. We took washed platelets, we treated the platelets with protease or anti-alpha-2 beta-1, and subjected to the binding to the collagen or fibrinogen or fibronectin coated to microtiter plates. And the adhered platelets were lysed and membrane acid phosphatase activity was measured. So in this case, you can see over here, so our protease and anti-alpha-2 beta-1 inhibited the platelet binding to the collagen. However, our protease or anti-alpha-2 beta-1 did not affect the fibrinogen binding or fibronectin binding. Thus, our protease and anti-alpha-2 beta-1 prevent binding of platelets to collagen, but not to fibrinogen or fibronectin. Now, does the protease bind alpha-2 beta-1 integrin or collagen or both? That was our next question. For that, what we did was, we took the protease, we subjected to STS page under both reducing and non-reducing condition, and native, that's NNPS, that's protease, as well as EDTA treated protease. So these are the resolved protease in STS page. So protease was resolved in STS page. It was electroplated to PVDF membrane, then incubated with platelet lysate. So in this case, we incubated with platelet lysate, then probed with anti-alpha-2 beta-1. Incubated with platelet lysate and probed with anti-alpha-2 beta-1. So this one we stripped off and incubated with collagen and then probed with anti-collagen antibodies. You can see over here. So in this case, you can see a signal over here, protein signal. So you can see clear-cut alpha-2 beta-1 integrin. It is not subjected to degradation or whatever. So that means our protease is binding alpha-2 beta-1, not the collagen. Our protease binds alpha-2 beta-1, but not collagen. Does the protease cleave alpha-2 beta-1 integrin or simply bind? So for that, we took washed platelets and treated the platelets with protease, and EDTA treated protease as well. We made a platelet lysate. We ran STS page. So this is the STS page pattern, and did Western blotting here, transferred to PVDF membrane, and probed with anti-alpha-2 beta-1, and then HRP anti-mouse antibodies. So here you can see over here, so the alpha-2 beta-1 integrin remained intact even with increasing concentration of our protease, remained intact. It is not cleaved. This is the loading control. So alpha-2 beta-1 integrin remained intact. So protease bind alpha-2 beta-1 integrin, but did not cleave it. This is the bottom line of this experiment. Then what is the effect of our protease on glycoprotein 6, which is another major collagen receptor. So for that, what we did was, we took the washed platelets and treated with the protease for 1 hour, and we made platelet lysate, and we did SDSPH, and electroblatted to PVDF membrane. We did Western blotting and probed with anti-glycoprotein 6 antibodies. So you can see over here, this is the control. So here the glycoprotein 6 is intact. There is no cleavage. As the protease concentration increases, you can see the cleavage of glycoprotein 6. And the cleaved product, you can visualize the lower, molecular size, the cleaved products over here. Thus, our protease cleave the glycoprotein 6, but it did not bind the glycoprotein 6. So we did the binding assay. So it just cleave the glycoprotein 6. Thus, protease bind alpha to beta 1 integrin and cleaves glycoprotein 6. So it establishes the contact place with the alpha to beta 1 integrin and then stretch itself and cleaves glycoprotein 6. So after this, what happens to the signal transduction? So we checked the signal transduction in general. We took washed platelets. We treated the platelets with protease and collagen. After 2 minutes, platelets were lysed, did STS-PAGE. We did 6 to 20% gradient gel here. This is the gradient gel we made. And so it transferred. So we did Western blotting, probed with phosphotyrosine antibodies over here. See, you can see over here, this is the control one, just stimulated with collagen, just collagen. You can see many bands which are phosphorylated and specific tyrosine residues. These are all the proteins, the intracellular proteins whose tyrosine residues are phosphorylated. You see the signal because we probed with phosphorylated tyrosine antibodies. And in presence of our protease, you can see that signal drastically decreases over here, suggesting that platelets or protease inhibits platelet activation and probably eventually degranulation also. So with this, we came to the probable mechanism of action of our protease and platelets. What platelets or what collagen does is, collagen binds to its receptors, alpha to beta 1 integrin and glycoprotein 6, and sends a signal. And that signal does two things. It is involved in degranulation. It promotes degranulation of granular substances. As I mentioned, platelets are loaded with 3 types of granules: alpha, dense, and lysosomes. Alpha granules, dense granules, and lysosomes. So the collagen, through a signal transduction, cause degranulation, and it also activates the receptor glycoprotein 2b3a complex. So in resting platelets, this receptor remain inactive. It cannot bind its ligands, basically the fibrinogen. But when it is activated, it changes its conformation. Now it binds its ligand. So this is the general mechanism of the collagen, how it induces platelet aggregation. But what our protease does is, it binds to alpha to beta 1 integrin of collagen receptor and cleaves glycoprotein 6. So it binds alpha to beta 1 integrin and cleaves glycoprotein 6. As a result, there is an impaired signal transduction. So as a result, no activation of the receptor, no degranulation. So finally, no aggregation of platelets. Thus, signaling, degranulation, and receptor activation is inhibited. No signaling, no degranulation, and no receptor activation, and finally, no aggregation. This is what the molecular mechanism we proposed based on our, some of the experiments that we did in our laboratory. So, in summary and conclusions, we have isolated a non-toxic and non-lethal protease. This protease inhibits blood coagulation, that removes clotable fibrinogen as well as dissolves whole blood and fibrin clots, inhibits platelet aggregation. As a result, this may be explored to treat diseases associated with vascular disorders or cardiovascular disorders. So this is my group. This work was done by Dr. Kumar. The entire work was done by Dr. Kumar. And isolation part was another guy did that long ago, Dr. Jagdish. And this is my current group working with me. I worked with Professor T.V. Basapagoda for my PhD. But before that, I was a student of Indian Institute of Science. I worked for PhD degree initially with Professor P.S. Sastry. And Professor Ram Rajashekaran, the ex-director of CFTRI, was my senior. He helped me a lot in all of my studies and all. Professor B.H.U. is my current senior in the department. He is my great friend, good colleague, and helped me in all respects. These are the fundings. And I also thank Professor Barry S. Scholar for donating anti-alpha 2 beta 1. So we received anti-alpha 2 beta 1 antibodies from Barry S. Scholar from Rockefeller University, New York. I really acknowledge him gratefully. So thank you very much.
Thank you, Professor Kumparaju. You have virtually converted this entire, I think, 250 people across the length and breadth of the country to a virtual classroom audience. So we are mesmerized, sir. And personally, I feel as a biochemist, a teacher in biochemistry, that another teacher in Mysore University can, from my specialty, can be such a wonderful teacher.
Yes, sir.
Can be such a wonderful teacher. So really, sir, you was very wonderful. You have explained every nitty-gritty questions. Now, before we open it to the audience for any questions, I would like to just showcase some of the points here for Professor Kemparaju's lecture here.
Yes, sir. Yes, sir. Yes, sir.
Yeah. So this is one case in time that where he has not studied only platelet, he has studied in parallel the protein part, thrombus part. So these two go side by side. When there is thrombosis, platelet aggregation, and clot formation, thrombus fibrin polymerization. They go side by side.
Sir, your voice is breaking, sir. It's not properly audible. I can't hear you properly. Sorry, sir. I can't hear you properly.
No, sir. So it is actually not for you. It is for the audience in large who are listening to this, younger people on the faculty. So what I again repeated, the Professor Kemparaj's talking showed that one can study platelet. If one wants to study thrombosis, one can study platelet part, cell signaling, as well as the protein part. How the clot formation and fibrin degradation and fibrinogenesis, fibrinolysis, these are to counterbalance enzymatic systems. One has to focus on. So very efficiently to study the toxins, he has studied both of us. That is one thing, take-home lesson for all the audience there. Number two, being in he himself admitted that he doesn't have so much genetic, but having said that, when publishing nature communications and the journals like that, so that's so that you see his experiments, he has focused mostly on HTS page, both gradient and homogeneous gels, maybe reducing, non-reducing both. So here, put the condition of HTS page as well as Western. So with these limitations and idea stretching, one can have very wonderful results out of that. And also wherever he has required that he is deficient of funding, he has requested anybody can do that. He has requested Gary Kohler, who is an eminent personality. Yeah, the requested for activity, they will be very generous to take it activity. You have to pay the postage, whatever, and blue dart service, etc., and they will pay that. We have done that earlier. So this is also one thing that how one can progress very consistently in the research. Thank you, sir, for motivating so many participants.
Thank you, sir.
And
Thank you, sir.
I request to stay a little longer so that if participant has some query, he can directly contact you. Tell you after here. Okay, over to the next one.
Yes, yes. Thank you, sir. Thank you, sir.
You have to tell, every participant should tell their name and place before they ask the question. That's a request.
Okay.
अनम्यूट कर दिया ना पार्टिसिपेंट अनम्यूट
Hello. So everything becomes silent now. I can't hear anything.
Very good morning, sir. I am Shwetanka Prasad from Manipal Tata Medical College, sir, Jamshedpur.
Madam.
Sir,
Madam.
I have
Good
one
morning, madam.
small
Good morning,
question.
madam. Yeah, you are audible.
Am I audible, sir?
Yes, madam, you are audible. Very much.
Sir, is there any correlation or association between the activated platelet and basophils in cancer cells, sir?
See, cancer cells, forget about just cancer cells. In general, actually, the activated platelets, they express a lot of P-selectin molecule. And even when they are enormously activated, they even shed the P-selectin molecule. So with the help of P-selectin, people have demonstrated, a group from Canada, they are working extensively the crosstalk between activated platelets and neutrophils. So the activated platelets eventually activate neutrophils. They have demonstrated that. That group is actively working on that. Yeah, maybe I think this area needs further investigation. It's open for people. Like many people can take up this kind of work and you can start working on. This is still in its infancy. See how the cells crosstalk take place. So people are now exploring the crosstalk between the activated platelets and the neutrophils. And people can try for basophils and other cells as well. It's a possibility.
Thank you so much, sir. Thank you.
Yes, ma'am. Yes, ma'am. Welcome.
Good morning, sir. Am I audible?
Yeah, you're audible.
Yeah. Thank you so much for a nice talk, sir. And my question is a little out of the box. As you mentioned that netosis, the process.
Yeah.
And this process, whether it can be influenced by the megakaryocyte, as you know that megakaryocyte is the big bone marrow cell.
Yeah.
So whenever studying the netosis, I mean, simultaneously, did you observe what will happening in the megakaryocyte biology?
No, that part so far people have not started. So
okay.
That thing, we don't get any kind of information.
Yes, sir.
This area is still, again, it is still a starting area. So it has to open up now. Maybe youngsters can take up this forward.
Yes, sir.
Bright chance, there's a bright scope for studying all the interactions. So netosis, the cells and all those things. See, the thing is that netosis is for only neutrophils. And cells other than neutrophils, if they participate the same way, that process is called etosis. And even megakaryocytes might end up in etosis as well. So you have to study.
Yes, sir. Yes, sir. Why? Because it's a very big, big bone marrow cell. So
multinucleate giant cell.
Yeah, multinucleate cell. Under this, it can grab the other cells basically.
Yes,
so,
exactly. Maybe.
Yeah. So in that context, it's very interesting, sir. And one more thing you
really
mentioned that. Yes, sir. And one more thing you mentioned, very interesting point, that generation of the megakaryocytes other than the bone marrow, it's a lung. And what could be the, the process how it will generate? You are thinking that.
That's what I too don't know. But
Yes,
I just gone through the article. But they also mentioned like how exactly megakaryocytes move from bone marrow and settle in lungs has not been
Yes,
clearly
sir.
understood. But they have observed
Okay.
using intravital
Yes.
microscopy technique. So we
Okay.
have
Okay,
intravital
sir.
microscopy, probably our next speaker is going to speak about that, intravital
Yes, sir.
microscopy. So
Yes,
using
yes.
intravital microscopy, they have demonstrated the release of platelets from the lungs. But
Okay.
how exactly megakaryocytes travel from bone marrow and settle in lungs has not been addressed properly.
And yeah, and one more, the last question, sir. As
Yes,
you are working on these poisonous compounds, I mean, can it applicable for translate to the human injection kind of thing?
oh,
Yeah,
sure. See,
yeah,
many of the
yeah.
toxins, they serve as
Okay,
prototypes for the drug discovery.
okay.
You know,
Right.
I take an example of your,
Yes, sir.
blood pressure. We have
Yeah, yeah,
print
yes, sir.
type
Yes,
of
sir.
medicines, captropil.
Okay. Yes.
Actually, the drive to make captropil was
Okay.
from a snake venom toxin.
Oh, yes.
Based on the
Okay.
structure of snake venom toxin, initially
Okay.
captropil was made. And
Oh,
from that, we have now the generics of captropil for blood
okay.
pressure control. So
Okay.
likewise, we have, they are leads for so many things.
Oh, and
For
my
example,
doubt is, so since it is a biological compound, maybe over a period of time it can, change its structure, can act as a epigenetic factor for other disorders. Nothing will happen. You are thinking that in
which one?
whenever enter an human physiological system.
You
So,
talk about
so
venoms.
yeah, venom snake. Yes, yes.
That part we have not studied. People have not studied
Okay,
in that line. But
okay.
anyway,
Yeah,
it's a foreign structure, and most
yes, sir.
likely proteinaceous in nature. Probably it is likely that they are getting degraded as the time
Okay.
passes on.
Yes, sir. Your study is very nice, really nice, sir. Thank you for information,
Thank you
sir.
very much.
Thank you so much.
Thank you.
Yes,
Thank you.
yes.
Good morning, sir. Sir,
Good morning.
it's Gaurahari Sahu from Central University of Punjab. Sir,
Fine.
in your earlier slides, you have described that we can inhibit the platelet or the thrombosis activity in two ways. One by inhibiting the activation of platelets, and another by inhibiting the fibrinogen kind of that process. So, in whole of your slides, you mainly focused on the inhibition of the formation of the binding of fibrinogen with two platelets. So my question is that in your earlier slides, you also have mentioned that we have used RGDS sequence, the 4 amino acid sequences, to
Three
mimic
acids.
to mimic the binding of fibrinogen, and that's why we have inhibited the aggregation of platelets. And in most aggregation studies, we also use RGDS sequence to inhibit the aggregation. So what are the shortcomings in using the RGDS sequences that are used by the medicines that we have come forward for new compounds to inhibit the aggregation?
I think I'm not getting your question. What is your actual question?
Sir, in one line, actually, if RGDS sequence can block the receptors for the binding of fibrinogen and other agonists, what are the shortcomings in that using that sequences so that we are uh, using more and more advanced, like you have used one protease to block the receptors. So, what are the shortcomings in that RGDA sequences by which we have came forward to decipher new things to prevent the receptors?
Shortcomings, I don't know exactly what are the shortcomings of that, but the things that bind to glycoprotein 2B3A complex, that is fibrinogen receptor, must have RGD sequence. So all your fibrinogen, fibronectin, vitronectin, they all possess RGD sequence or KGD sequence. So without RGD or KGD sequence, they cannot bind to the receptor. So people capitalized that part and made synthetic structures. So I mentioned a couple of drugs over there. Those are all derived from the disintegrants that were isolated from snake venoms and created that sort of drugs, and they block the fibrinogen receptor. But the shortcomings, I really not aware of that. What could be the shortcomings and all? Or you talk about maybe side effects. Side effects, I really don't know.
No, sir. I was actually asking about that. If only the mimicking of a RGTS sequence can do all those things, then why we have to go for a multiple sequence of the inhibitors? That's what I was asking.
Oh, see, RGD sequence is actually the structure that is the motif that binds. But to keep the motif, you need another structure, is it not? Just RGD is not enough. Just RGD is not enough. To have a motif, to have a protein motif structure, you need to have a little bigger structure. Then only you can keep the RGD, so that it can interact with the receptor molecule. You need a protein. The entire the rest of the protein structure to hold that RGD sequence. Just tripeptide will not inhibit. You need a protein motif.
Yes, answered.
Okay, thank you.
Now he's satisfied. So now department Professor Mishra is here. He will talk to you for Dr. Gepard's lecture. Thank you, sir.
I'm not audible.
Nice lecture, sir. Very
Voice is
interesting. Very
voice is
interesting lecture, sir.
Voice is breaking.
Voice is.
I can't hear you.
Okay. Okay, sir. Very good afternoon, sir. And the lecture was very nice. And
Good afternoon, sir.
this was interactive session, sir. Very सब लोग interact किए, very interactive. And
Thank you, sir.
all participants.
Thank you, sir. Thank you very much.
Thank you, sir.
Thank you very much. Thank you very much, sir. Thank you. Thank you, Das, sir. Yes, sir. Thank you. Thank you very much. Thank you, everybody. Thank you.
Thank you. We shall be in touch. And perhaps next year we are meeting in Manaus. So
Yes, sir.
now Deepavali is coming, so please seek some blessings from Mother Chamundeshwari for our
Yes, sir. I'll pray. Definitely. Thank you, sir. Thank you very much.
Be here online, no problem. Now, just now you tell our foresight over here, if you like it, channel. No question. Yeah.
I can't. Your voice is breaking, sir.
ये हमारे ईयरफोन में भी ब्रेक हो रहा है क्या?
You can be online also.
Yes, sir. I will be in on.
Yeah, yes.
Yes, sir. Yes, sir.
यहाँ वही ब्रेक है. नहीं.
It's perfectly.
हाँ. फिर अभी ब्रेक कर रहे हैं या फिर कल किया था?
That means that is like all participants for a small break. We will be meeting exactly at 12:10 for Dr. Jyotsna Kailash's lecture on flow cytometry. After that, there will be a virtual lab experience for all of you. Flow cytometry topic itself. So we'll be waiting for just 5 minutes, then rejoin again on 12:10. Thank you. But they can make sure I want to ask you a question. Yeah, please utilize this time if you want to. Then you can put all your queries in the chat box, so that we can respond to them at the earliest. Thank you. Professor Temperature is there on the side also. He's sitting there. So if you have still more some queries there, so you can post your queries. So we shall try to respond to you. So, Professor Temperature, the basic premise of this workshop is that I want you. Are you listening to me? No.
I'm muted.
It's not clear. It's not clear.
We are able to hear you, sir, but the voice is not clear.
Yeah, I can hear the sound, but voice is not clear.
Not to worry. We are trying to rectify.
सर देखो ये जो है तो इसका लेक्चर हमारी तरफ से होगा ना
Yes, sir. Anyway, thank you very much.
Dr.
Thank you very much.
Kamlesh, kindly check the mic. There's some disturbance.
Okay.
sure, thank you for the suggestion.
Thank you very much. Thank you.
अच्छा
उसके क्या करने बोलो तो कुछ क्लास
इज देयर एनी क्वेश्चन
यहाँ नहीं सुनाई दे रहा बोलो तो
इसमें सुनाई देगा ना
इसमें नहीं आ रहा है
उसमें आएगा ना
हेडफोन में नहीं
यहाँ पे बंद होगा यहाँ पे क्या मैं टर्न ऑन कर दीजिए बाकी लोग म्यूट करिए
नहीं सुनाई
हेलो आई एम ऑडिबल ठीक Is there any voice breakage issues? यदि आपको voice break हो रहा है तो आप हमें प्लीज उसमें बता सकते हैं. Okay, so that we will correct the system again because we will be utilizing this small short break for the correction of system. We hope that our voice is not. किसमें अच्छा हाँ इस दूसरा कर लीजिए वो भी सही है ये इसके ऊपर दूसरा कर लीजिए इसके ऊपर सही है मैम बोल रहे हैं
माइक माइक स्पीकर दो ना
इको ही हो रहा है इसमें इको आ रहा है इको आ रहा है अभी में हाँ टोटल एकदम सही है ये वाला ब्लूटूथ है क्या तो इसको बंद कर दीजिए हेलो इज द वॉइस ब्रेकिंग राइट नाउ कुछ इतना सब चूहा घूम रहा है तो किसने किसने कुछ गड़बड़ किया है
Dr. Kamlesh, kindly speak.
Am I audible right now? Is there any voice breakage?
Yeah, no, it's clear now.
Okay, thank
नो
you.
क्लियर सर
It's clear.
Yeah, thank you. Please.
मिल गया सर ईमेल?
Yeah. So, am I audible? Yeah. So, as yes, yes. Okay. So, we'll be resuming our session again. So, for this session, our speaker will be Dr. Josna Kailashia. She is Associate Professor in Department of Biochemistry I.M.S. B.H.U. She has done her MD and PhD from the our department itself, and she is a faculty in the department since 2015. And her area expertise is platelet functions and signaling, nanomedicine, newer drug delivery methods for anti-cancer therapies, stem cell tissue research, biosensors, and medical and clinical biochemistry and techniques. She has received awards like Shamlal Saxena Memorial Award, NMS Amritsar Award, Order of Academic Excellence in Publication, and also she has granted one patent. She has till now 23 publications in different reputed international and national journals. So I welcome Dr. Josna. She'll be enlightening us on the topic of flow cytometry or FACS. Thank you.
Thank you, Dr. Kamlesh, for this introduction. And hello, everyone. Good afternoon. So I'll be talking about the application of flow cytometry in study of platelet functions and signaling. So first of all, I'll tell you only a small introduction about flow cytometry. Then I'll tell you about platelets and what are the functions or signals you can detect in platelets by using flow cytometry. Okay. Then I'll show you some of our papers from our lab that in which we have used these techniques. And then I'll show you video of our experiments, how we do it. So I'll be here with you till the experimental part also. So, okay. So we can go ahead. ये move नहीं कर रहा है. Okay. So something about flow cytometry. So if you are a beginner, so you should know what is flow cytometry. If you're expert in flow cytometry, then it's very good. You'll understand it clearly. So flow cytometry is a method of measuring multiple physical and chemical characteristics of any particle or cell by optical or electrical means while sample is flowing inside the instrument. That instrument is flow cytometer. Okay. So why we call it flow cytometry? Because sample is flowing inside the instrument, and while it is flowing, we are measuring some of the characteristics of those particles or cells. Those characteristics can be physical or chemical. Okay. So by flow cytometry, we can do qualitative as well as quantitative analysis. If you have any question, you can simply type it in chat box. I will entertain it later. Okay. So flow cytometers can measure relative size of the particles or cell by flows forward scatter of, or we call it simply FSC, or it can measure relative granularity or internal complexity of the cell or the particles. We call it side scatter or SSC. And then we can also measure relative fluorescence intensity. It depends on what fluorophore you have added to your sample. So depending on that fluorophore, you can measure fluorescence intensity. And this fluorophore will depend on your instrument setup. And there can be so many variations in flow cytometers. There can be 6 channel, there can be 16 channel, there can be 18 channel flow cytometers. So you have to just look for details of your flow cytometer that is available in either your lab, or if you have any central facility, you can look there, or even it is possible to outsource flow cytometry experiments also. So you can simply prepare your samples and give it to that other lab facilities and get it analyzed in flow cytometers. Okay. So these are the parameters, 3 parameters that you can use in flow cytometry. Forward scatter, that is for relative size. Side scatter, that is for granularity or internal complexity of the particles. And third is fluorescence intensity, depending on whatever fluorophore you have added to the sample. Okay. So uses of flow cytometers. There are so many uses of flow cytometer. You can detect surface markers of cell. For example, in leukemia diagnosis, they use extensive panel of antibodies for leukemia identification. Then you can also detect so many intracellular factors also, like DNA content, mitochondria, or ROS levels. You can detect intracellularly. And so there are so many possibilities of using flow cytometers in research as well as in clinical use. So these are some examples of applications of flow cytometry, like cell counting. Yesterday we showed you one cell counter from Beckman Coulter multisizer 4. That is a kind of flow cytometer. So we use it for cell count only. It is not having fluorescence detectors, so we cannot do other functions with it. So cell count or particle count, cell size, relative cell size, every flow cytometer will tell you. But absolute cell size can also be measured by properly calibrating the instrument with the fixed size bead. So that is also possible. Then cytoplasmic granularity you can detect. Then cell surface antigens or phenotyping of the cell can be done with flow cytometers. The apoptosis analysis can also be done. Intracellular cytokine levels, production of different proteins, and other signalings can also be detected by flow cytometer, both quantitative and qualitatively. Then gene reporters, fluorescent gene can also be tested by flow cytometry. Then cell cycle DNA content analysis, cell proliferation, cell sorting, these all can be done by flow cytometers. Then parts of an usual optical flow cytometers are 4: fluidic system, laser system, detectors, and electronics or display units. Okay, fluidic system is what it is composed of different tubes and sample insertion port where you place your sample, then it will go and flow inside the system. Then there will be a laser interrogation of your flowing particles. So it will constitute of different lasers, different colored lasers, different wavelength lasers. Then there are detectors. After cells have been interrogated by lasers, the signals are generated, and those signals are detected by different kinds of detectors or photomultiplier tubes. So it depends on your instrument setting, what kind of and how many numbers of detectors you have. And then electronic display. So simply it is computer in your software. So there are so many types of the flow cytometers, and so they are compatible with their own software, and we have those computers where those softwares are installed, and we have to use those computers connected to the flow cytometer for analysis and display part. Okay, so this is very simple explanation. I'm not going to details about the engineering part and how these detectors work or laser system works, because for today's experiment we don't need that. So this is simple diagrammatic presentation of a flow system inside the flow cytometer. So here you can see we have a mixture of particles or cell as a sample. When we put this sample in your flow cell or sample insertion port of the flow cytometer, it will create a flow like this, and cells are arranged in a single cell file manner. Okay, because it is very important actually. So to prepare sample correctly for flow cytometer, if your sample is too crowded or too concentrated, this pattern, single cell file pattern, will not be achieved. Okay, so you have to take care about this. That is why I have placed this diagram, so that it gives your impression in your mind, so that you prepare the sample correctly and well diluted. Okay, if your sample is too concentrated, the cells will be crowded and your data will be wrong. Okay, so here samples are flowing. One cell will come in front of laser beam at a time. Okay, two cells should not interact with laser at a time for correct flow cytometry experiment, right? So here you can see this yellow cell has come in front of the light beam, that is incident laser beam. Then this cell will generate some signal because of its size because of its internal complexity, and also if you have stained the sample with something, either a dye or an fluorescent tagged antibody, that signal will be generated from this point, and this signal will go to different kind of detectors. You can see forward scatter detector, side scatter detector, FITC detector. FITC is a fluorescent probe or fluorophore, and another is P detector. This arrangement of flow cytometer depends on your instrument type. It can be different in different instrument, but usually most common instruments are having this setup only: forward scatter, side scatter, FITC, PE, PERCP, and APCE channels. Okay, so 6 channels and 4 fluorophore can be detected in a cell at a time. Okay, so this is simple instrument setting. Then detectors will detect the intensity or fluorescent intensity or signal generated from the particle or cell. Then this signal will go to your electronic display where software is installed. The software will analyze or see the data, and it will display the data. I'll show you in experiment part also. So there are different software setting also you have to learn for your own particular instrument. But in general, settings are forward side scatter and side scatter plots, scatter plots, histogram plots. So in that way, you can display your data. Okay. Yes. So again, I'm placing this diagram because you have to remember in flow cytometry experiment, your sample should be single cell suspension and well diluted to form a single cell file line in the flow system. If it is too concentrated or too diluted, your experiment will be wrong. Okay, so again, remember this. So this is simple protocol for running a flow cytometer experiment. You can see here this is sample. Your sample, it should be single cell suspension. And then you can simply label your cells or their function with either a dye, fluorophore dye, or any antibody tagged with fluorescent probe. Okay, so there will be a labeling step. After labeling, you have to give some incubation time. It depends on what labeling reagent you have used. Then after incubation, there can be other steps like permeabilization of cell or fixation of cell. So it depends on your particular reagent. For some reagents, you have to fix the cells and then permeabilize them to make the dye or probe get inside the cells. So that depends completely on your particular experiment. But in general, this is the commonest protocol. You take your sample, you add your dye or fluorophore, then you incubate, then simply after dilution you can run in the flow cytometer. So here you can see here it is giving example of 2 dyes. One is appearing red and one is appearing blue. Okay. So one is appearing blue. So here you can see what is happening inside the flow cell at the interrogation point. Interrogation point, what is interrogation point? The point where cell is hit by an incident laser. Okay. So there it generates signal. Here you can see blue particle is generating blue signal, and red particle, when it comes, it will generate red signal. Because this particle is white in front of red laser, it won't generate any signal because it is not having any red color dye in it. So it won't give any signal. But blue particle will give signal in blue laser, and red particle will give signal in red laser only. Okay, so this is how the specificity of the fluorophore is maintained, and you will see color of only one single dye at a time. Okay, but in plots, but in software plotting, you can display multiple colors or multiple gating. I'll show you later. Okay, so this is how the analysis is done, and this is how your scatter plot will appear. There are so many precautions and limitations and steps you should follow before running flow cytometry experiments, right? So first is instrument calibration. Whatever instrument you're having or maintaining, it is having its own steps for instrument calibration and quality control. You always have to perform it to see that instrument is working fine so that your data is reliable. Okay. Every time you run flow cytometer, you have to do instrument calibration and quality control. Okay. It depends on that instrument and the manufacturer's guidelines. Some companies say that at least do quality control and calibration once a week. Some say, okay, you have to do it daily if you're running experiments daily. Okay. So it depends on your instrument. Then while sample preparation, you have to make single cell suspensions. Okay. You cannot run aggregated samples. You cannot run clotted samples. You cannot run pieces of tissue. Never, never. Otherwise, it will block your instrument and it will be a great loss. So single cell suspension is very important. So again, I want to remind you about yesterday's experiment, that how platelet preparations were done. We were adding platelet inhibitors at almost every step so that they do not spontaneously aggregate with each other. Okay, if there is even small number of platelets that aggregate in your sample while preparation of the sample, then it will completely ruin your data. Especially in flow cytometry, it can even damage the instrument. So that sample preparation step is very important. Then correct getting and settings. You have to learn which population you have to select for analysis. Okay, I'll show you gating in experimental part, and then you have to apply correct settings. You must know what fluorophore you have added, in which channel or in which FL detector they will show results. So that those settings you have to apply. Okay. Then choice of fluorophore. There are so many fluorophores. It depends on what function or what surface expression you want to analyze or see. Okay, so those depends on availability also. Some companies have actually most of the antibody companies are having different platelet related antibodies or markers, so you can search, but your fluorophore of choice should be whatever is compatible with your instrument. You cannot have any fluorophore that is not available with your detector. If detector is not available for you in your instrument for that particular dye, you cannot use it. So it depends on your experiment also and your instrument setup also. Then you have to always take fluorescence related precautions, like you have to protect those fluorescent dyes from light exposure. Okay, otherwise their fluorescence will quench and you will not get enough strong signals. Okay, then after running and before running also, there should be proper cleaning of flow system or your instrument. If you don't clean your instrument, if you don't maintain your instrument according to manufacturers, then your data, first your data is not reliable, and then your instrument can also get damaged. Okay, so these are the precautions you have to maintain. Otherwise, flow cytometry experiments are very easy, very easy. But the tough part is maintenance of those instruments. Okay. If you have any question, please type in the chat box. After the end of this lecture, I will address them. Then now something about platelets. So since yesterday we are talking about platelets and so many information has been already given to you. I will not go into detail, but simply these platelets are fragments of megakaryocyte that circulate in blood. They get activated whenever there is any agonist present, whenever there is injury, whenever there is stimulation, by even by stress stimulation, platelets can get activated. Platelets are inherently CD41 positive. So if you are analyzing a mixed cell sample, you can tag your platelets by CD41 antibody. Okay, then main function of those platelets are to stop bleeding. Whenever there is injury, whenever there is break in blood vessel, platelets function is to stop the bleeding. But behind that, in the background, so many things are happening in the platelets. Manas sir told us today, then Kemparaju sir also told us today what signalings are there. So many surface receptors are exposed, so many intracellular signalings are there. So you can target all those functions and signals by flow cytometry. Okay. So this is just another so that you can remember what is happening. This I took this photo from Google only, so you can see there also. So these are these are resting platelets, simple, calm, doing nothing, discoid shape, smooth surface. They simply flow in the system, your blood vessel. But whenever there is agonist exposure or injury or any stimulation of platelets, they quickly change their shape. They are throwing so many these projections. They release microparticles. So their phenotype abruptly changes from resting to activated platelets. So this is from microscopy, but some of the signals of activated platelets can be seen in flow cytometry also. So many changes are happening in platelets when they get activated, like surface expression of so many receptors and protein changes, like membrane content can change, surface receptors can change. Intracellularly, so many changes are also happening, like ROS levels may change, some enzymes get activated, granules are released, calcium, intracellular calcium levels are increased. And then there are some metabolic changes, like Manas sir already told you. There are so many metabolic changes, especially related to energy consumption, right? So these are the changes happening in the platelets when they get activated from resting stage. So all of these you can target. The only condition is they should be tagged with something fluorescent. Okay. If it is surface expression is changing, you can simply give antibody or surface binding specific protein or something that can bind to that expressed receptor. So this will show you changes in the receptor expression. Then intracellularly, you can target so many things. You can see mitochondrial function. I will show you this experiment, how to analyze mitochondrial functions in resting and activated platelets. Then you can see ROS levels. It is known that activated platelets are consuming so many ATPs and utilizing energy pathways. So it leads to increased ROS production also. So that is also visible in activated platelets. You can tag enzymes also. For that, you have to permeabilize the cells, then add something that will bind to the enzyme. Especially kits are available. So many kits are available for flow cytometry. You can simply Google it, and it will show you. With so many companies, you can target those enzymes like caspase. Then granule release can be detected. Yesterday we showed you how to see granule release by aggregation, luminescence aggregation, but it can also be detected by flow cytometry. Simply we do p-selectin staining, because whenever there is degranulation, p-selectin exposure on the platelet surface is increased. Okay, intracellular calcium levels can be detected because we have so many dyes for detection of intracellular calcium. In our lab, we use fluo4. Okay, so fluo4 AM. So fluo4 dye is specially for calcium detection. We add it in the sample. The dye goes inside. When it binds with intracellular calcium, it gives you increased fluorescence. So these are the methods, simple methods by which you can target analysis of so many different platelet functions and signaling. Again, these things have been discussed already today also. So what is happening? This is blood vessel layer. And whenever there is injury in the blood vessel, there will be exposure of collagen. then these platelets get exposed to collagen, and this collagen is also a known agonist or activator of platelets. So here you can see the platelets are having smooth surface, nothing so special about this. But once they get exposed to collagen, so many receptors are exposed. So these are receptors which mark activation of platelets, and you can target these receptors for knowing if the platelet is in active phase or in resting phase by flow cytometry. Okay, so once these receptors are exposed, so many signalings are there, inside out, outside in, then platelets will change their shapes, then throw so many projections, and there are so many interactions, like with ADP, they will release ADP, von Willebrand factor, thrombexin A2, fibrinogen. Because of degranulation, granules will be thrown out and they will release these molecules. Then what is happening? At later stage, so many other platelets are recruited at that site. They interact with each other, interact with fibrinogen, and they make a clump, right? And their final function, what is visible to us, is formation of plug that will close this injured site, right? So in between, so many things are happening from this A to E, okay? So we have so many phenotypes and receptor activations, exposures, and intracellular signaling that we can target for analysis during flow cytometry, right? So many molecules, so many receptors are there. So I'll show you two examples, flow cytometry experiments. And this is a simple diagram of what activated platelets look like. Okay, not exactly, but this is only a schematic diagram. So you can see there are collagen receptors, there are von Willebrand factor receptors, thrombin receptors. Here you can see alpha 2b beta 3 receptor, that is fibrinogen binding receptor, or we can call it integrin 2b3a. Okay. So in resting phase, these integrins are in closed format. You can see here in the left, they are closed like this. There is no cavity, there is no binding site in those receptors. But once they come in contact with any platelet agonist, or when the platelets are activated, these receptors will become open or activated. So from here, they will open like this. So when they open, you can see a little cleft is made in this. So this cleft is containing binding site for the fibrinogen. Okay. So here, when platelets are activated, these receptors will change their conformation. They are protein receptors, so proteins can change conformation. When there is conformation change, the binding site for fibrinogen will be exposed. Then fibrinogen can go and bind in that cleft, and this fibrinogen will interact with other cells. So all the cells will clump together and make a clot or aggregate. Okay. So this receptor expression can be detected by flow cytometry. So this is the one I will show you. Here it may look complicated, but during experiment, it is actually very easy. So I have kept the experiment at the beginners level so that everyone can understand. Then you can do multiplexing of those experiments also. So again, the receptor level diagram, what changes are happening in the receptor level after activation of platelets. You can see here, these are same integrin alpha 2b beta 3 receptor in the closed format, closed conformation. But when they are activated by thrombin or any other agonist, the conformation will change and that binding site will open. Then so many other receptors can be visible in the activated platelets, like P-selectin. P-selectins are usually intracellular when the platelets are in resting phase, but they come outside in activated platelets. So you can target P-selectin also. Anti-P-selectin antibodies are there. Then there are granule release. Here you can see there are granule release. So content of the granules will be thrown outside of the platelets that can be detected. So yesterday chemiluminescence method was shown to you for this degranulation assay. But in flow cytometry, we usually target this p-selectin. But other granule markers can also be used here. Then there will be change in membrane, platelet membrane, phospholipid orientation. Okay, phosphatidylserine usually is inside the cell. It is facing inside the cell. You can see here, this is phosphatidylserine. But when platelets are activated, this orientation will just flip. There are flippase and proppase enzymes which will flip these phosphatidylserine from inside inner leaflet of the platelet to outside, that is outer leaflet of the platelet. So phosphatidylserine will be exposed outside in platelet, activated platelet, also in apoptosis, apoptotic platelet and other cells also. It is this annexin 5 is not specific for platelet, but you can use this marker for activated and apoptotic platelet because this annexin 5 protein binds to uh, outside expressed phosphatidylserine. So this is another marker you can use for platelet function. So this is how we go for surface expressions. Okay. Then there are some inherited platelet receptor defects where these are very rare disease. We usually don't get them. But if any clinician is suspecting, okay, there may be the patient's bleeding disorder is not getting, they are not getting in good response from the patient by treatment, they suspect about this inherited disorder. So in these disorders, some platelets are inherently deficient in, some receptors are inherently deficient in platelet surface. That is why platelets cannot work properly. So examples are Glanzmann thrombocythemia, where alpha 2b beta 3 receptor, those are fibrinogen binding receptors. They are deficient or maybe absent. So they cannot properly bind with fibrinogen, and that is why they cannot properly aggregate. Similarly, Bernard-Collier syndrome is having low glycoprotein 1b95 or low von Willebrand factor binding receptors. So again, their aggregation is defective, especially in presence of ristocetin. So this flow cytometry technique can aid to your diagnosis of these disorders by aggregation. First, we usually test these diseases by aggregation by putting so many different agonists in the aggregation tube, and we see what agonist is working and what agonist is not working. But if you point out, okay, maybe this is the defect, maybe this is the disease, you can confirm it by flow cytometry by testing the surface expression of these antibodies. They will surely be less than the healthy person. So, okay, so here flow cytometry can be used for clinical experiments also. Then intracellular signals. Till now I've shown you so many surface expression changes, but there are so many intracellular signals also that can be detected by flow cytometry, like increased mitochondrial respiration or membrane potential. It usually happens in activated platelets because they are using so much energy, they're consuming so much energy, so their mitochondria are also very active, so their membrane potential is usually high. Okay, so this is one way you can do this experiment, or you can, if you want to test a drug, if it is damaging platelets or if it is damaging mitochondria, again you can use this mitochondrial membrane potential experiment. I will show you this experiment today. Then intracellular ROS generation can be detected. There are so many dyes available which interact with nascent oxygen and they convert to fluorescent molecule. And this fluorescent molecule concentration or level can be detected by flow cytometry. Then intracellular calcium, I told you already, flow 4 is a very good dye that will bind to calcium. It will give a fluorescent signal. So these are the some examples of intracellular signaling you can use. If whatever you are interested in it is not in the list, you can simply Google it and see if those experiments are compatible with flow cytometry. Usually there are so many experiments and so many reagents available. Then there can be some other type of experiments in platelets that will show you if your molecule of interest or if your nanomaterial is damaging to the platelets or if it is not damaging to the platelets. Okay, so for those experiments you can detect platelet damage or apoptosis signals by an exin 5 binding assay, mitochondrial damage again you can see, or caspase activation assays are also available for flow cytometry. The kits are available. It is very easy. All the instructions are given with the reagents. You simply have to follow them. So now I'm showing you some of our publication from our lab where I have extensively used flow cytometer for my experiment. So this is the first paper. I worked on this in my MD time where we invented a biosensor to detect platelet-derived microparticles. So those platelet-derived microparticles are small fragments from the platelets which are released when platelets are activated. Okay, so these are kind of marker of thrombus risk or procoagulant state in blood. So we made a biosensor for that, but to confirm the function of biosensor or effectivity of biosensor, we had to had a standard equipment. So flow cytometer was used as standard equipment to check proper function of biosensor. So here you can see the A graph is showing data from flow cytometry. You can see scatter plot of flow cytometer. Platelet-derived microparticles are here, red marked, because I have stained them with antibody, PAC1 antibody. These microparticles are PAC1 positive. PAC1 antibodies bind to alpha-2b beta-3 integrins also, the same integrins where fibrinogen is binding. Okay, so you can see here in resting platelet samples, there are so few microparticles, but when we activate the platelets, those number of microparticles increase. You can see in R1 upper right and lower right quadrants. Okay, so this is data from flow cytometer, and this was used as standard for detection of microparticles, and it was corroborated with the biosensor. So I won't go in the detail, but you can analyze microparticles also with flow cytometers. And again, the comparison of flow cytometry and biosensor. So you can see the pattern is almost same. The samples, healthy subjects who showed low number of microparticles in blood by flow cytometry also showed less signal in biosensor. And patients, acute myocardial infarction patient, that is heart attack patient, showed higher number of platelet-derived microparticles through flow cytometry. They also showed higher signal in biosensor. So that was our work. Then next paper where we used flow cytometry was this one, engineered human platelet-derived microparticles as natural vector for targeted drug delivery. In this work, we loaded platelet-derived microparticles with some fluorescent drugs or other molecules, and we had to see if that there is successful loading. So this successful loading was confirmed by flow cytometer. Here you can use flow cytometer for this purpose also. So here you can see doxorubicin was the drug because it is very brightly fluorescent. So here you can see without addition of doxorubicin, this is very low signal, and when we add doxorubicin, the particles are getting doxorubicin and they are loaded with doxorubicin, the signal is very high. So this confirmed drug loading into microparticle by flow cytometer. So this is another application of flow cytometer. Then this is my most recent paper. Here, flow cytometry was used for nanomedicine purpose. So the title was Effects of Nanoceria on Human Platelet Function and Blood Coagulation. So nanoceria are serum oxide nanoparticles. They are somehow present in the environment through petroleum products and some cosmetic products. So we are slowly getting exposed to them. So we wanted to test if these nanoparticles affect platelets or not. So this was the study, and flow cytometry was one of the techniques I used. I used so many techniques. You can see yesterday's experiment, that is platelet aggregation. So serum nanoparticles showed inhibition of platelet aggregation. Then main application of flow cytometer was to see if nanoceria, usually in engineering and other applications, they are claiming that nanoceria are having antioxidant property, okay, ROS quenching property. So I wanted to see if they really have antioxidant property, especially in platelets. So this was my experiment with flow cytometers. You need special kind of flow cytometer where time parameter is also there. So you can see real time data of your treatment and changes. So here you can see these are these dots. These are scatter plots only. And these dots, each dot is presenting one platelet. Okay. So I run resting platelets here, then add H2O2 or any other signal, any other ROS generating stimulus, then this ROS signal is visibly increased. This ROS signal is due to DCHFDA. This is dye that will show you ROS signal. So here we used flow cytometry. Then intracellular, another function of platelet was observed in this paper, that is intracellular actin polymerization. Okay, so intracellular actin polymerization detection is also possible. So you can refer to this paper for detailed experiment. Today I'm not going to explain these ones. I'll show you simple 2 experiments only. So this is detailed experiment what we did. This left side we are having without platelet, without treatment with nanoceria particles, and here we are seeing platelets with nanoceria particle. You can see when we add thrombin, when we are adding thrombin, this point, this arrow is showing addition of thrombin in the sample. You can see 40% of the population is in upper right quadrant. It means they are positive for ROS signal. But in nanoceria, NC600 is nanoceria with concentration 600. So in nanoceria samples, this concentration or the count of platelets or percentage of platelets which are ROS or reactive oxygen species positive are only 32. So from 40%, they come to 32% only when we give nanoceria. So it confirmed that nanoceria is actually having antioxidant properties inside platelets. Same experiment was repeated with H2O2, and you can see without nanoceria, positive events are 76%, and with nanoceria, they are 73%. So slight inhibition is there. Now we can come to workshop experiments. What will show you for today's experiment. So I have planned 2 experiments. First experiment, I will show you how to stain surface expression of any receptor. Okay. So for today's experiment, I have targeted alpha 2b beta 3 integrin that will bind to fibrinogen. So the experiment is expression of fibrinogen binding receptors on platelets. Okay. The second experiment is something about intracellular signaling, whatever is happening inside the cells. So for that, I have used mitochondrial membrane potential staining by MitoTracker dye. It is easily available. You can purchase it. It is a membrane permeable dye. It will reach mitochondria inside the platelets, and healthy mitochondria usually accumulate this dye, and damaged mitochondria will not accumulate this dye. So this is the first experiment. I have outlined simple steps. You can either take a screenshot, or these steps are available with the your reagent also. If you purchase MitoTracker, they will have information about that dye, and they will give you details of how to stain your cells. Maybe you may need a little bit modification or standardization for your laboratory, but usually the steps are same. So in first experiment, that is platelet surface fibrinogen binding receptor expression assay. The aim is to compare expression of fibrinogen binding receptors, those are integrin alpha 2b beta 3, in resting platelets and in activated human platelets. Okay, sample preparation steps are first we take washed platelets. Those sample preparation steps we already showed you yesterday, how to use differential centrifugation for preparation of washed platelet from whole human blood. Okay, the count is usually taken 1 to 2 into 10 power 8 platelets per ml. Usually you can reduce the count also, it works fine, because for flow cytometry we need very small volume and some few number of cells, like 10,000 cells gated are required. So, okay, so it will be enough. Then you divide those samples in your tube, different tubes, depends on how many samples you want to keep. Then because we are comparing resting with activated, you have to activate one sample with a thrombin. Okay, so you usually use 1 unit thrombin per ml. You can standardize with smaller dose also, sometimes 0.5 also works. क्या हो गया ये?
Decline करो.
मेरी तरफ से? Sorry for the interference. Okay, thank you. So this thrombin dose is very variable. It depends on age of thrombin. Sometimes we get very new thrombin stock and it works fine. Even 0.1 unit will work. Sometimes it gets old. With time it degrades maybe, and its activity is also that those times we need 1 unit per ml. So it depends on so many factors. If you don't keep thrombin in proper storage, then again its activity will decline. Okay, so because I wanted to be sure that all the platelets are activated, I used highest dose. Okay, 1 unit per ml, but maybe in your lab 0.1 will also work fine. So we will add thrombin in one vial, and that vial will be labeled activated. Then we have to incubate the sample at 37 degrees Celsius for optimum temperature. 37 is our body temperature, optimum body temperature. So most of the bodily reactions happen at 37 degrees, so we have to mimic them. So we'll keep those samples at 37 degrees Celsius water bath for 10 to 20 minutes. This thrombin actually activates platelets very quickly, within seconds. You can see in the aggregometer aggregation experiment that after adding thrombin, the graph falls very quickly. So it happens within 2 to 4 minutes, but we give them time, these samples some time, like 10 to 20 minutes. Even 10 minutes will be enough to complete the surface expression. Okay, conformational changes. Then after incubation, we'll add your fluorophore or dye or anything that will tag your special surface expression or receptor with something fluorescent. So here I have used fibrinogen because these receptors are fibrinogen. I have used commercially available fibrinogen that is already tagged with the fluorophore, that is Alexa 488, right? So whenever these fluorescent fibrinogen molecules will bind to platelet, it will give us signal in flow cytometer. Okay. Then after adding the fluorophore, we have to gently mix it. Always mix platelet very gently, है ना? Vortex जाके नहीं, मतलब you should never vortex your live platelet samples. Then mix gently, and then again we have to keep it in water bath for optimum antigen antibody reaction or surface interaction. Then after 20 to 30 minutes, the sample will be ready for experiment. है ना?
Who is this Dharmendra Kumar sir? Please don't try to intervene between the presentation. Because constantly your request to annotate is being followed. Yesterday also, I think the same request has been done by the same user. Please don't do it.
Okay, if you have any doubt, any query, or any even objections, you can type in chat box. I'll go through the chat box after this talk, okay? So, okay. So this was a simple protocol for surface expression. You can stain for surface receptor. By this method, only your probe will change. If you are targeting CD41, then you'll add tagged CD41 antibody at the step number 5. Okay, so this protocol will work for any surface expression experiment. Only you have to change the fluorophore. Then second experiment will be for something intracellular, that is mitochondrial function. So mitochondria are required for cellular respiration. So if platelets are healthy, mitochondria are healthy. If platelets are damaged or dying, mitochondria are also dying and they are damaged. So this whole function is based on that. And when platelets are hyperactive or activated, then mitochondrial functions will also increase. So it is based on this property of this permeable dye, that is mitotracker, and this mitotracker will get inside mitochondria and it will give fluorescent signal. So aim of this experiment will be to compare mitochondrial functions in resting and activated human platelets. Sample preparation steps are almost the same, We'll take wash platelets, we'll divide it in different vials. We usually use 1.5 ml Eppendorf tubes. Then here I am using one control, another control, that is CCCP. This CCCP is known disruptor of mitochondrial membrane potential. We know that CCCP damages mitochondria, so we'll keep it as control. Then we have to give treatment with thrombin like previous experiment, incubate at 37 degree for at least 5 to 10 minutes. In 5 to 10 minutes, all the effects of thrombin and CCCP will be visible. Then after this incubation, we'll add mitotracker dye to all the tubes. We'll mix it gently. Then we'll keep it again in water bath for 20 to 30 minutes to let the mitochondria accumulate all the mitotracker dye. Then after this incubation period, we can dilute the samples and run in flow cytometry. There can be other few changes in the protocol if you are targeting something else inside the cells, okay, like you want to target some intracellular protein. So usually your antibody is not permeable, will not cross the cell membrane, it will not go inside unless you permeabilize the cell. So you can simply add a fixation, formaldehyde fixation or paraformaldehyde fixation, and triton permeabilization step in between before adding that antibody. Then antibody will go inside and it will bind to your target protein and it will give you signal. So small modifications are possible, should be done according to your experiment or your aim. And usually those protocols in general are given with your reagent data sheet. Okay, so always go through reagent data sheet, always see in which solvent you have to mix, at what temperature you have to keep those. Okay, if those should be kept at simple fridge or at -80, how to thaw it. These are all specific requirements. So always read a data sheet of your reagents. Okay. So mitotracker dye, I already told you, we use mitotracker red. It is red fluorescent dye. It will stain mitochondria in live cells and healthy cells. And it depends on mitochondrial membrane potential, that is proton throwing out of internal membrane. Okay, so there is usually a proton gradient in healthy mitochondria and healthy cells. So this depends on that only. If that proton gradient or mitochondrial membrane potential is damaged, the dye will not be accumulated well enough in the mitochondria. Then CCCP, I told you, it is control. It is used as control. It is known mitochondrial membrane potential disruptor. If we add it in healthy cells, it will damage membrane potential, and we'll see it as reduced signal of mitotracker. I'll show you in video. So please play the video.
Questions
Okay. Okay. I can address some questions now before playing the video.
Yeah.
कितने बज गए?
1 बज गया।
मैंने 1 घंटा बोल दिया। ओहो। Wait, pause.
And open the chat box.
क्या है?
ये unmute कर दीजिए, कोई question नहीं।
All these possibilities with flow cytometry can be done in a single go, means single experiment for multiple parameters. Yes, it can go in a single experiment, single tube only. It depends on your fluorophore. Your fluorophore should be different for different channels. If you have 4-channel flow cytometer, then you can add 4 different fluorophores at a time and you can analyze at a time. If you have 16 color flow cytometer, then you can add 16 different fluorophores at a time and analyze them. It is possible.
Madam, good afternoon.
Good afternoon.
Ma'am, thank you for that query. And I am very excited to hear about this technique. I was not aware of this flow cytometer, anyways, with multiple possibilities. I have 2 questions. First thing is that how will you compare chemiluminescence method, which was addressed by the previous speaker yesterday, and the flow cytometer? I feel flow cytometer is much superior technique, and you can do multiple both surface markers and changes in the internal environment of the cell. Multiple possibilities are there. Apart from that, what are the differences with respect to
Chemiluminescence and flow cytometer you want to compare.
Yes.
Actually, it completely depends on your research question and whatever is available to you. Yesterday, we showed you chemiluminescence release of ATP,
Yes.
right?
Yes.
While aggregation. So that ATP release is, you can see it in real time while you add your agonist, right? So
All
you
right.
can parallel with the aggregation. That is not possible in flow cytometer, right?
Okay.
It will not be. But flow cytometer itself has so many other advantages, like you can target so many things in a single sample only.
Yeah,
So
I am looking at that. I wanted to see certain receptor, trafficking or receptor expression and all that on the surface of the cell. So I think flow cytometer will be the better way. So my second question is now definitely it will be a very expensive equipment.
you
So
can always outsource it.
yeah, that is my query. I mean, how much do you think probably for a sample? I know it depends on parameters, but on an average, a sample, how much?
And I can tell you about my university central facility. University one, not the institute one. University is having facilities for internal candidates. I think it is 400 to 500 per 10 sample or per hour. In 1 hour, we can go for 10 samples.
Okay.
And it will be 500 rupees per user. Okay. For external, I think it will be a little bit costlier, like 800 to 1000 per hour or per 10 sample. So this is the usual cost, but there is another way you can collaborate with labs who have this equipment. Okay,
I am
collaboration.
speaking from Kalyani. I mean, what is the? I mean, you know, how can we collaborate? I mean, I am speaking from West Bengal. How will the sample, can be transported? Or,
I'll tell you that. Okay, good. So there are so many ways samples can be transported, and most preferred one is fixed sample for flow cytometry.
Okay.
0.2%
No.
formaldehyde fixation. No, you can read about it.
Okay.
Those samples can be run in flow cytometers even after 7 days. They are good if your fluorophore is compatible with the fixation. Okay, you can collaboration is very simple. You can simply see who is having this facility. You can talk to lab in charge or the person who is handling those instruments. If your work and interest match, then you can arrange for sample transports very easily. Or your students can come here with the samples. We sometimes do that. We sometimes go. I myself went to A.I.M.S. Delhi for my samples in past, so it is possible.
Okay. Thank you, man. Thank you so much.
Yeah. Nice talk, man. Can I ask the question,
You all can,
Yes.
switch on your camera so I can see whom I'm talking to. Otherwise, I feel like I'm talking to the speaker.
I have some video issue over here. I just want to ask my question.
Problem, it is up to you.
Yeah. So as you mentioned that it's very interesting that you done the microparticle identification through FACS analysis, right?
Yes. Yes.
So what kind of methodology you followed? Like, I mean, since the microparticle can be released by any kind of blood cell.
Okay.
So you are saying that platelets only release the microparticles. We analyzed that. How do you know that they are releasing from only platelets?
Okay, good. This question should always come in mind before starting the experiment, that how can I make sure that those particles are from platelet origin only. So in background knowledge, we know that most of the microparticles circulating in blood are from platelet origin only. Okay. The next thing is, what are the markers? I have to think like what are the markers that will only stain platelet dry microparticles and not any other microparticles, right?
Yes, yes.
Will be making it more specific. So the diagram I showed you, it was stained with the PAC1 antibody. That stains for integrin alpha 2b beta 3, and it is specific for platelet derived microparticles only. So only those particles will be having PAC1 antibody positive, other will be having negative. In the diagram, did you see this green particle?
I understand. I saw that.
Those particles were negative for PAC1, so those particles were from other cell origin. I completely could exclude those by getting.
Okay. And one more thing, so you want to extrapolate this microparticle is like, combination kind of drugs or any the platelet disorders furtherly? And
No, please ask again. I couldn't understand.
no, this microparticles and all, once you identified that, I mean, obviously they will change in the, control versus platelets. I mean, in our experiment. So, I mean, how you extrapolate for the biology of human system?
Human system, I already told you, blood is mixture of so many things. So many particles are there from other cell origin. In blood, we cannot isolate platelet microparticles. Other even sorting was failed. We tried it. No, in A.I.M.S also, in other where they have sorters. So sorters cannot separate them, but in flow cytometry we can target and identify their origin. Okay, but when you want to have pure preparation of platelet derived microparticles, so first you have to prepare pure suspension of platelets. Okay, when you have pure platelet suspension, then you activate them. So in supernatant, you will be having only microparticles of platelet origin. So that is another approach.
And one more last question, madam. So I think this microparticle is, really based on this size matter actually.
Yes.
Yeah.
Yes.
So how did you maintain that, microparticle size instead of like they were further converted into exosomes, other kind of particles also, no?
No, we cannot maintain. No, we cannot tell platelets that only, okay, I want microparticles, not the exosome. They will do their work. When they are activated, they will throw out all of those, all various size ranges of those.
Yes.
Right?
Based on
We
size
cannot.
also we call it as another name. So that's why, yeah.
Exactly. So I have seen some papers that have characterized different particles generated by different stimulus. Maybe you can search it. So different platelet agonists actually can affect the what particles they are releasing. So you can go for that. But in our lab, we do not usually distinguish them. And another part is your flow cytometer. Their detection range is usually less than either 300 or 100 nanometer. So below that, they are actually not seeing anything. Everything else is debris. So our flow cytometer is having lower limit of 300 nanometer. So we can see only above that. So all the exosomes are excluded, right?
Yeah. Okay. Yeah.
Anything else?
Yeah. Thank you so much, ma'am. Yeah.
Welcome.
Hello.
This
is Ivanala Hadam from A.M.S. Kalyani.
Hello.
Thank you for the presentation. Ma'am, actually, my question is,
Mhm.
flow cytometer is used like for single cell suspension, no?
Yes.
So when you activate the platelets, then they aggregate.
No, we
Then
don't let
will
them aggregate. We don't. We don't move them or stir them or shake them. That is important.
so that
And
means
no, we don't let them aggregate.
But I thought that is like one of the basic property that once there are stages, no, that you have mentioned, then
Huh.
it
The count should be in a flow that they do not spontaneously interact. That is why I emphasized on platelet count or your sample count earlier. You know, it should be very less so that platelet do not get crammed up. And another thing is you never stir your sample after adding thrombin. If you stir or you shake, gentle tapping is okay. If you shake, then they will aggregate. Otherwise, they don't. You can see in experiment, thrombin stimulated platelets are also showing the same forward and side scatter, same relative size and granularity. Their aggregates are not there. If you shake them, if you add calcium, calcium is important. Never give calcium to thrombin treated platelets, otherwise they will make clump. Okay, so we avoid thrombin. Another precaution we take is we add BSA in the buffer, sample preparation buffer. Yesterday we told you, right?
yes,
BSA.
yes, yes.
So it will
Okay,
prevent the aggregation.
okay, thank you.
So we are actually running out of time. We'll just play video. You can again start chatting, typing your question in chat box so I can address it later. Please play the video. क्या करना है? Off हो गया? So I am going to show you two types of staining for flow cytometry in platelet functions. So first is fibrinogen binding in activated platelets, and another is mitotracer staining or mitochondrial membrane potential staining in platelets. So first I'll start with washed platelets. This is washed platelets. First, fibrinogen binding in washed platelets. ये प्रेजेंटेशन पे चल रहा है ना? So I am going to show you two types of staining for flow cytometry in platelet functions. So first is fibrinogen binding in activated platelets, and another is mitotracer staining or mitochondrial membrane potential staining in platelets. So first I'll start with washed platelets. This is washed platelets. First I'll prepare samples for fluorescent fibrinogen binding in washed platelets. So I'm taking two tubes. I'll mark them. बहुत लंबा हो गया unexpectedly जबकि मैंने स्लाइड बहुत कम बनाई. Okay. इसको ऑफ कर दो ना. So I have taken two tubes. I'll mark them. One is resting platelets, and another sample will be activated with thrombin. So I'll write it thrombin. Okay. So these are both for fibrinogen staining. I'm marking them fibrinogen. Okay. Then I'll take 50 microliter each in both tubes, 50 microliter washed platelet samples in both tubes. So I don't have to mix anything in resting platelets because I have to keep them in resting phase. But I'll activate the thrombin activated sample with 1 unit thrombin. So this is 1 unit thrombin. So concentration, working concentration I'm using is 1 unit thrombin per ml. So I'll add 0.5 microliter from this stock. I'll gently mix this tube and then I'll keep it in 37 degrees Celsius water bath for 10 minutes. So I'll bring it there, then I'll come back after 10 minutes incubation at 37 degrees Celsius. Okay, so 10 minutes incubation at 37 degrees Celsius is over. I have two tubes. One is not activated, one sample platelet is not activated, and another sample, the second sample, is thrombin activated platelets. So in thrombin activated platelets, there is increased binding of fibrinogen. So fibrinogen binding receptors expression, surface expression is increased. So fibrinogen will give fluorescent fibrinogen in the both samples. So resting platelet will not show increased fibrinogen binding, but thrombin activated sample will show increased fibrinogen binding. So next step will be staining with fluorescent fibrinogen. So we have Alexa 488 tagged fluorescent fibrinogen. I'll just add it, but before that I have to switch off lights because it is the fluorescent tagged reagent. So I'll switch off light, then I'll mix it. So here I have fluorescent fibrinogen. Working concentration will be 10 microgram per ml in each tubes. So for that I'll add 0.33 microliter in both tubes for 50 microliter sample volume. Gentle pipetting or mixing. I put back the stock covered. Now I'll gently mix both samples so that my reagent, my dye is evenly mixed in samples. Gentle tapping will be enough. You don't have to shake or vortex the sample because vortexing will be activating to the platelets. Okay, so one should avoid vortexing live platelet samples. Now staining reagents are added. I have to cover both the samples with the foil to prevent light exposure. There. Okay, both samples are ready, but before flow cytometry analysis, I have to incubate both for 20 to 30 minutes again at 37 degrees Celsius. So I'll keep both samples in 37 degrees Celsius, and after incubation, the sample will be ready for flow cytometry analysis. Alright, I'll go and keep it at 37. Now I will start staining for our second experiment, that is measurement of intracellular mitochondrial membrane potential in platelets. Okay. So this experiment will be done by using MitoTracker dye. It is commercially available and it gives fluorescence in FL2 and FL3 channels. I'll show you in flow cytometry. Right now I'll show you how to prepare samples. So I'll take 3 tubes. One will be again RP, resting platelets without any treatment. You can call them control resting platelets. Second sample will be thrombin activated platelets. And third sample will be our positive control treated with CCCP. CCCP is known to cause reduction or damage of mitochondrial membrane potential, so we use it as positive control. All 3 tubes are marked. Now I'll dispense wash platelet sample in each tube. So I'll take 50 microliter each. You can run samples with 100 microliter or 200 microliter sample volume also, but because flow cytometry is very efficient, so I can do analysis with very small volume. I'll only need 10,000 gated platelets. I'll show you in analysis part. So small sample volume is also enough for flow cytometry. So I'll take 50 microliter in each tube. Some amount around 100 microliter of wash platelet is remaining. I'll keep it as unstained sample. Okay, you should not throw away this unstained, because unstained sample will be required for gating in platelet sample analysis. Okay, so this is unstained remaining wash platelet sample. I am keeping it away for now. I'll reuse it for flow cytometric gating. Okay, now I'll show you staining of mitotracker dye. First, I'll give treatment of CCCP in this tube. Alright. Working concentration of CCCP is 100 micromolar. So I'll add 0.25 microliter for 50 microliter sample. Working concentration is 100 micromolar CCCP. So CCCP sample is treated. Now I'll give treatment to this thrombin sample, thrombin activated platelet sample. Again, 0.5 microliter, that is 1 unit per ml, final concentration of thrombin. So platelet treatment is complete. Now I have to incubate these samples at 37 degrees Celsius for at least 5 minutes, so that effects of thrombin and CCCP will be visible. So I'll go and keep them in 37 degrees Celsius for 5 minutes. 5 minutes incubation of the sample is over. Now I can stain these samples with mitotracker dye. Mitotracker dye informs us about mitochondrial health. It means there will always be some mitotracker signal in normal platelets, baseline signal, but when CCCP or some damaging mitochondrial damage is there or CCCP treatment is there, mitochondrial membrane potential actually decreases. So mitotracker dye does not get accumulated in the mitochondria, inside the mitochondria, so signal is decreased. When there is thrombin stimulation or activation of platelet, there will be hyperactivity of mitochondria, so there will be increased accumulation of mitotracker dye inside the mitochondria. So now in next step, I will stain these samples with mitotracker dye. So working concentration of mitotracker dye will be 0.5 micromolar in each sample. So for that, I'll add 0.25 microliter of mitotracker stock solution in each sample. So here's my mitotracker dye. Because this dye is again fluorescent, so I have to switch off lights. Then I'll add this dye to the samples. I've added mitotracker in all three samples. I'll put it back. Now I have to mix all tubes by gentle tapping at the bottom. Because it is light sensitive dye, I have to protect all the tubes, so I am covering them with aluminum foil. All tubes are covered, so I can switch on lights now. Alright, so these are for 20 to 30 minutes. By that time, our first experimental sample that was fluorescent fibrinogen staining will be ready. So after keeping it in 37, I'll go to my flow cytometer and I'll start it. So we can start analysis of previous experimental sample, okay? And this is its sample insertion port or SIP. I have to use these tubes they are compatible with this BD-FAX caliber to put my sample in. So this is how I put my sample. I remove the previous tube, then I insert my sample tube. Okay. Then the whole operation of this machine is through software. I'll show you the software in the next step. You can use any flow cytometer that is having forward side scatter and different fluorescent channel like FITC channel, PI channel, PERCP channel, APC channel. So all those flow cytometers will work with this experiment. I am showing you with our machine BD-FAX caliber. We have other flow cytometers also, they are also compatible. So 6-color flow cytometers work well with these experiments. Cytometry analysis. So I'll open the software. Here, it is open. Now I have to plot scatter plot to visualize my sample, so visualize platelets. So first I'll draw a forward and side scatter, then fluorescent channel. Okay. So first I have to do the settings. Different flow cytometers can have different softwares and different operational method overall settings. So it depends on what kind of flow cytometers you have. So this particular software is for BD-FAX caliber. So we do the settings accordingly. So I'll adjust it to acquisition and analysis. There. So first scatter plot will be forward and side scatter. I can increase the size. You can see meter. There, it is connected to cytometer. Then this tab will show me when to acquire or when to stop analysis. Okay. The next are small particles, small cells. I have to set all channels in log scale, not in linear scale. So if you are analyzing other cells like WBCs or cancer cells, larger cells, you have to set FSC and SSC or forward and side scatter to linear channel. Okay, but all the fluorescent signals will be in log channels only. So here I am setting FL1 channel. So I'll adjust it to forward scatter, that is size, and FL channel, that is for fluorescent tagged fibrinogen. So Alexa floor signal will come in FL1 channel. So this is how my setting is done. I can also add additional graphs like histogram. I'll add one histogram for FL channel. This is histogram plot. And I want it to be of FL1 channel. I have to make a directory or I have to make a file so that my sample will be saved there. So I am saving it for now in desktop. I have my own folder. There I've saved one folder. And in workshop, I'm showing you two experiments. So first experiment is of fluorescent fibrinogen. So I'm adding another folder, fluorescent. So all my fluorescent fibrinogen samples will be saved in this folder only. Okay, I'm prepared with my samples for analysis. So this is how setting is done. Always better to dilute the samples before flow cytometry so that single cell file can be achieved and there is no overcrowding during flow cytometry analysis. Okay, so I'll go and dilute my samples. So here are my two samples, one resting and one thrombin activated. Already saved about 100 microliter of washed platelet without any treatment and without any staining. So I'll take about 50 microliter of this unstained platelets and I'll mark this new tube unstained. I'll take 50 microliter of washed platelet, unstained washed platelets into this. Then I'll dilute this sample with pack sheet. Okay. Here. So fact sheets is provided by BD for our instrument. For different flow cytometers, there can be different sheet solutions. So it depends on what kind of flow cytometers you have. So you have to use its own compatible sheet solution. So I'm using this one. I'll add 200 microliter of fact sheet solution in my unstained sample. There. So now it is diluted and now it can be run in flow cytometer. I'll gently mix it. Then I have to also dilute my other stained samples before running in flow cytometer, but because they are fluorescently tagged, I'll do it after switching off the lights. Okay. I'll dilute both these samples similarly, adding 200 microliters of fact sheet solution in 50 microliter sample. Cytometry analysis. I'll keep the lights off so that I can run these samples because they are fluorescently tagged. But before just putting the samples, you have to switch on and wash and process your flow cytometer according to the manufacturer's guidelines. So I have already done that. I have switched on my flow cytometer. I have done all the washing steps and purging steps. Now my flow cytometer is ready to use. Whatever flow cytometer you are using, stick to the guidelines for starting the equipment and taking care of the equipment. Proper washing should be there. Then only you should put your samples for analysis. So let's go to the flow cytometer for running the samples. But before putting it in SIP or in flow cytometer for analysis, I have to transfer this volume into this kind of tubes. This tube. Okay, this is compatible for fax caliper. You can use different tubes for different flow cytometers. I told you we have to save the file name so that your sample data will be saved in that file name. I have already made directory and folder for saving my data so that I know where that data is going. Now I have to save file name because I have placed unstained sample. I'll just name it whatever your sample is or whatever codes you use. This one, I use this one, unstained wash platelets. It is subjective. Okay? You can give your own name. Now this one is ready. First we have to, for running, we have to tell the equipment which particles or which cells to analyze. For this I have to use setup setting and I'll just run acquire so that I'll know where my cell population is appearing. So I'll run acquire for few seconds. Here you can see, here this is my wash platelet samples. Okay, so I'll just pause, then I'll apply gating. Gating is nothing but telling the instrument telling the instrument which population to analyze. Okay, so I'll just draw a gate for my population of interest. So this is free gating tool. This is my platelet population. The other unwanted data, unwanted noise and smaller particle fragments, they all will be excluded. So this is how we do gating. Now I have to tell the instrument to exclude all the data outside platelet range. For this, I'll do setup, acquisition and storage. Then I tell the equipment that accept only this G1 gate. This gate name is G1 or gate 1. So I'll tell the instrument that collect data only in G1 gating range, and now this event count option is there. So I want to count or I want to analyze 10,000 platelets per sample. So my setting is already placed there, 10,000. You can increase or decrease this number if you want to analyze 50,000 samples. Then you have to just change this to 50,000, so that the equipment or the software will run sample till 50,000 events or 50,000 cells of your interest are analyzed. For now, for platelets, we generally do 10,000. So I'm putting this number 10,000 platelets or 10,000 cells in G1 or gate 1 region. Then the flow cytometer analysis will stop. Okay, okay. So that gating is applied. Now I'm setting abort so that previous setting events are gone or removed because I don't want them. Now I want to run my sample according to my gating and my settings only. Okay, so I have set all the parameters, all the gating here. Now I will remove this setup setting so that if setup setting is clicked, then their data will not be saved. Okay, so I am removing this setup. Then I will simply click acquire. So this is how wash platelet unstained sample has been acquired. You can see this is the population of interest. This is its FL1 signal in the plot. So now I can remove the sample, put sheath in it and put the cytometer in standby mode till I run my second sample. Now I can do one more thing. I can apply a quadrant gate here. Can you see? Yes, I have applied quadrant gate. Shift from left to right. So this is how gating is done in scatter plots, and quadrant gating is placed in scatter plot. Put it in these tubes for analysis. So my second sample is resting platelets without treatment with thrombin. Instrument is in standby, so it's not running now. And before running, I have to give name to this this sample, that is, I'll change it. So here I have given name to this sample, add and save. Now I am putting this flow cytometer in run mode, and I'll acquire the data with this tab here. Okay, so data has been acquired. It is very quick. Again, I put my flow cytometer into standby mode till I put my next sample. Am I audible?
Yeah.
Okay, so I've shown you how each sample is acquired and data is saved. So we can simply skip those part. I have more samples, but those will be run in the similar manner. First, you dilute it with 200 to 300 microliter of sheath. Then you put name in your this data so that you can track that or find that data later, and it is saved in that name, and then you simply keep running those samples. Okay, so I'm just skipping that part so that we can save some time. So this, if you can see, this data is for thrombin activated sample. You can easily see the shift in FL1 channel. I'll show you this data in my slides also, so how you can interpret. But right now I'm just skipping with the running part, okay? Because those are the similar ones. All the samples will be run in the similar fashion. Only thing is you have to set the detector or the FL channel correctly. For fibrinogen experiment, I have set FL1 channel, and for mitotracker, I can set FL2 or FL3 channel. All other steps will be similar. Okay, so this is for mitotracker. You can see in FL2 channel, the same diluted samples. Okay, I think I can play the last. Now I'm placing this sample, CCCP sample in flow. Last one is CCCC sample, CCCP. Okay. So then I'll just click acquire. Here. Okay, so very quickly 10,000 platelets data is acquired and flow cytometer stops, and now we can go further. Because this was like my last sample, I am removing the sample and I will put sheath in the SIP. From here we can start shutting down procedure of the flow cytometer if you are done for the day and you don't have to run any flow cytometer sample or flow sample. So you can start shutting down the flow cytometer. The procedure will depend on your guidelines, manufacturer guidelines. So we'll follow the guidelines provided by BD for fax caliber, and we can shut down the equipment, but we'll keep this software open for analysis because. Okay. So you can stop the video and resume the PowerPoint. PowerPoint जो था उसको खोल दीजिए वापस, पेन ड्राइव में होगा. Lastly, I have to show you how the data interpretation is done, because I've seen new students struggling with this. Share नहीं है? Share कीजिए. I've seen some new students or the students who are new to flow cytometry on how to actually interpret the data and how to present the data in your thesis or maybe in your paper. So I think that part should also be covered with it, because it is little bit different than other experiment, because the graphs are peculiar for flow cytometers. Okay, reading the graph is also a skill. So here we come to data analysis and presentation part. This is the last part. I hope I'll finish it early. So here, the first experiment was expression of fibrinogen binding receptors or integrin alpha 2b beta 3. You can see this upper left graph is for resting platelets without thrombin activation, and this lower right part is for thrombin activated platelets. So you can see the population is shifting from left to right. You can see the scale, log scale in FL1 channel. You have to read it in x direction, okay, x axis. So you can see the population completely shifts from left to right. So it means that all these right lower quadrant population is positive for fibrinogen binding receptors or activated. These are activated platelets. Okay, so almost 94% platelets are positive after thrombin exposure, thrombin treatment. So it confirms that thrombin treatment stimulates the platelets and makes their integrin receptor activated. Okay, so this is one way of presenting data. This is a kind of raw data. You have to show it in your paper that this is how the plots were appearing. Okay. Then another way is through histogram plots or overlay. This is histogram overlay. You can show single graph also, because we have used 3 samples. I have just prepared overlay. All the flow cytometers are having their own software, and they are having their way of making overlay. It can be different for other flow cytometers, but most of the flow cytometers I've seen, I think all of the flow cytometers I've seen will give you this overlay histogram. So you can see in this plot we have mean fluorescence intensity plotted in x-axis, okay, and number of platelets in y-axis. So you can see in unstained platelets the signal mean fluorescence intensity signal is very low here in black one. When we stain them with fluorescent fibrinogen, some of the platelets are showing receptor expression, but when we activate these samples with thrombin, all the platelets have shifted to right side. It means all of them are exposing fibrinogen binding receptor in their active conformation, right? So this is how you present the data. And this is mitochondrial membrane potential experiment. You can see we were having 4 tubes. One is resting. It is showing no signal, almost no signal in FL3 channel. It can be done in FL2 channel also. Mito tracker comes in both channels. And this green one is resting platelets. They are having healthy mitochondria. So they are showing some signal. And when you activate these platelets with thrombin, the graph little bit to the right. It means some of the platelets are having very, not some, you can actually you cannot actually say how many, but platelets are hyperactivated. That is why their mitochondria are hyperactivated and they are accumulating more of the mitotracheidine. That is why there is shift to the left side. And the control was CCCP. I told you already, CCCP will decrease mitochondrial membrane potential, so that you can see this blue graph shift to the left. It means their fluorescence signal has decreased. Okay, so this is how you present data. And you cannot just simply do one experiment and show in your paper that, okay, I have done this. No, you have to repeat your experiments at least five, 6 times. So when you repeat, you will have mean values and standard deviation. So you can plot in either in tables or you can show these bar diagram. Are we having next session on statistics or are we skipping to tomorrow?
Well, what is the time?
Those, yeah.
Okay.
If we can finish it in 20-30 minutes, we can have it.
Yeah.
Okay. So I'm showing you this because the next session, planned next session is about how to present these data in paper according to statistics analysis. Okay, proper analysis. So this is how you can done, because I have done only once, so I cannot show you standard deviation, but you have to show standard deviation after repeating the experiment. So this is for fibrinogen binding data, surface expression. You can see how much it is increased after thrombin activation, and this is for mitochondrial function. Okay, increase after thrombin, decrease after CCCP treatment, right? So other aspects, there are so many aspects of flow cytometry. Once you start using it, you will know how to go about this sample preparation. And there are, one student asked if they can do in one tube, one multicolor. Okay, you can do. Actually, this is preferred because it will decrease your efforts. But there are so many other things that you should think about. Why? You should think about before planning multicolor staining experiments. Like your instrument should be compatible. You should have fluorophore, compatible fluorophore. And then you have to go for panel designing. Most of the antibody providers are now giving free service of panel designing. And you should learn about compensation, because fluorophores are sometimes tricky. You know, if you regularly use flow cytometer, then you must be knowing. Otherwise, for new people, you should actually learn about these compensation and how to manage these. And then multiple gating strategies can be applied for positive, negative, double positive, double negative population. So it depends on your experimental design only. So many possibilities are there. Okay, that was my last slide. If you
Unmute all.
okay, so all questions are invited, and if there is no time left today, you can simply email me your questions. I will try to answer it, because I think I took so much time for my lecture. Sorry for
Madam,
that.
very good afternoon. I'm Shwetanka Prasad from Manipal Tata Medical College, madam. Madam, in the last slide, you said 5 or 6 times we have to repeat the experiment. So it is per sample, right, madam?
No, not for per sample. No, I'm not understanding. One sample you can run once, but you have to repeat the sample in another human being, another blood donor, or another mice, or another subject.
Sure, madam. Sure. Thank you, madam.
Okay.
Okay. I must thank Dr. Josna for her elaborate presentation on this flow cytometry part. She has taken pain to document everything, just like if you have been here, then whatever you have been witnessing, the same thing she has compacted in her video. So definitely, I think most of you must have been benefited with that. If you have any questions, then
Chat box, there are having
Yeah.
some question. I
Yeah.
can simply. Okay. Anyone who has not received their answer to their query can ask through mic.
I have one question for all the participants that we were planning to have one more session today regarding the statistics for this biological experiments. It will be around 20-25 minutes presentation. So if all of you agree, then only we'll proceed with this one, or if not, then we can proceed this with tomorrow's session. So I just need your affirmation for that. How many participants?
Yeah, type in chat box if you wish to continue with the next lecture.
Yeah.
Or you can
I
verbally.
have one question.
Oh, you have question, please.
I'm from IIT B.H.U. My name is Vishnu Priya.
Anyone come in this experiments, you can come to biochemistry department, find me, and you can discuss in detail about flow cytometry. Okay, I'm here only.
Okay, ma'am. Thanks.
You can ask now also. You can come to me also if you are in B.H.U only.
Okay, ma'am. I will meet you, ma'am. Thank you.
You can meet me. Then we can discuss in detail also. Okay, some participants have typed that we can continue.
Yeah.
2 participants. Lunch break. Okay, mixed. We are getting mixed signals.
काम ये कर सकते हैं कि एकदम छोटा एकदम समझ में भी आना चाहिए एक्चुअली फटीग होता है
I just request all the participants here. I hope many of you are here only. First of all, I must tell you that tomorrow's first presentation is on the practical part, unlike the 2 days that we are being having right now. So previously we will be having a theory on the first part and then practical session on the second part. But tomorrow that is a practical studied session with and also the presenter will be joining us from USA. So I request all of you to be in time for the first session because as I can see from today's data that many of you have joined quite late in the session. So you might miss the first session, which is quite practical from your point of view. So I request all of you to join dot on time at 10 a.m. so that we can finish the practical session. There will be 2 practical sessions tomorrow. So it will be easy for all of you to comprehend the things tomorrow. So I just request all of you to join in time at 10 a.m. So that we can finish in time and also you'll be able to listen to all the important session tomorrow. Thank you. So we'll be signing off now, and we'll be continuing with the biostatistic session tomorrow only. Okay. Thank you.
Bye, everyone. Bye.
Thank you, sir. Thank you, ma'am.
Okay. Yeah, bye.